Welcome to an on-the-road edition of the Demystify Sci podcast. We are headed down to Austin, Texas for our very first conference that is on April 7th and 8th. And as we travel, we are recording some conversations with ourselves, and we are recording some conversations also with our friends that we've been meaning to sit down with and talk about their theories with. And today we have Michael Hughes, who found us. I'm not exactly sure how
I
he
think
found us.
via Kurt's channel, actually.
Probably via Kurt. So thank you, Kurt, for maintaining an excellent channel over at Theories of Everything that is getting people to find us as well. And Michael's really into several topics that we've explored on the show before, which is structured water, which is the line between rationalism and mysticism, and the dark matter of science. Like, how are the pieces that we don't know about yet influencing the phenomena that we are observing, and so are having to kind of piece together in the dark how all of these unknown factors are influencing the observations that we make, both in the laboratory and out in the world.
Yeah, more specifically, I think most of today's conversation circled around the increasingly important active role of water in biological systems. So in the body, in the cells, how the water, which is generally thought of as sort of the invisible medium of the body, right? It's the background, the space upon which all of the molecules do their dances. It seems like there's a huge paradigm shift occurring right now in basic biophysics where that water is actually doing a lot of the activity. And it opens the door, so Michael and I were both at APS last, I guess it was almost 2 weeks ago now, in Minnesota, and we were talking a lot about water, but it also opened up these discussions about consciousness and about how all of these, how these experiences that we have we've been unable to pin them down to just the simple neuronal brain activities. And it ties into Michael Levin's research. I've had Michael on the show a number of times. This idea that conscious experience is distributed throughout the body, right? Throughout the cells. The cells have their own goals and desires, and there's communities of these. And the idea that there might be some role for the medium itself, right? Because water actually has a lot of the characteristics that we would normally ascribe to a computer like our brain, right? There's plenty of room for structural switching, for circuit behavior, electrodynamics, electrostatics. There's
the storage of information.
Yeah, in general, just processing of information, so computational power. And the more we learn about water's active role in the body, the more shocking it is that we actually have a landscape for information processing available. So that kicks open this entire can of worms, and I was kind of excited to find that Michael was starting to really get into the deep mythological and historical considerations about how conscious behaviors are distributed throughout the human project, but also throughout the cosmos, particularly through mythology in the past. And this leads us up to Rudolph Steiner and Christian mysticism, Western esotericism, these different framings for what it means to be a conscious entity in the universe.
Which seems really disparately related. Like, if you're like, this is a conversation about structured water and how water stores information, and it's also a conversation about Christian esotericism and mythology. But I think that Michael's making the point, the overarching point, that look, we have a ton of data that says that kind of weird, inexplicable things are happening in the body. Everything from near-death experiences to children being born with the memories of past lives, organ transplants carrying around personality, UFO encounters over the course of the last century, previous to that encounters with angels and whatever contextual creatures were of that time. But the problem is that studying those things is really difficult, because when you sit down and you try to study remote viewing, you start to get a lot of weird data, and the data behaves strangely. And so Michael's overarching point is, he's like, look, there is an electrically sensitive solvent in which all of this is happening.
Which can form structure, which does form very complex structures that are dynamic and change in response to environmental cues. much like a biological organism.
And those structures appear to inform the structure and function of everything else. Like we talked during the conversation, like, okay, so we have a vision of biology as being something that is membranes and proteins and these macromolecules banging up against one another to do stuff. But you can also look at that exact same picture and it's inverse and say that, okay, well, actually, it's the organizing principle of the water that is giving shape to all of these things that are happening inside of it. And so if you change the water, you change everything else. And if the water is electromagnetically sensitive, what happens when the magnetic field of the Earth reverses? Or there's a massive coronal mass ejection? Or there's something that comes out of the center of the Milky Way, some kind of cosmic ray burst. Like all of these things will affect
or
the
just
system.
what you eat, right?
Or what you eat.
Like just
So
really
much more
prosaic,
prosaically,
mundane
yeah.
things, right? So, I mean, I don't think that I know we didn't really get too deep into the historic treatment of these ideas, and there's a lot more to talk about there. So we're going to do another podcast soon and really start cracking open some of the mythological traditions as they relate to these basic principles, because that is something we've been studying a lot lately, and it's really fascinating. There seems to be a deep and rich history of people across the globe trying to make sense of these inexplicable phenomena and coming up with really interesting stories. And the basic underlying principle of that is, look, when you investigate something, you bring a metaphysical stance to it. This is something we've been talking about a lot on the show. You either acknowledge your metaphysical stance, that is, and this metaphysical being synonymous in some sense with your spiritual bent on things, right? You either acknowledge that, or you try to pretend like you don't have one at all, which is basically a lie. And so by acknowledging it and studying it and actually becoming aware and sort of taking the power back from it and really taking possession of your metaphysical bent, then you can actually maybe adjust where your theories, what kind of goods your theories are capable of producing in the long run. So we'll get more into that next time. But it's a really, really cool friend that we've made, and it's a really promising field of research. And I think we can learn a lot from water about the whole universe, actually.
Yeah, definitely. And so if you like what we do, come over to patreon.com, support us for a couple dollars a month. We could not do it without the patrons that we already have, so thank you to everyone who supports us. There's a list of all of those people at the beginning of each episode, and I just cannot say thank you enough. If you've been listening for a while and you're kind of like, I kind of want more of this, support us at patreon.com/demystifysci. $1 or $2 a month changes the world for us, and if it's something that you can afford, we would be absolutely eternally grateful. In return, you get both episodes early. You get them on Saturdays instead of having to wait for Sunday and Thursday, and you also get to join our patron chat where we get together once a week and we talk about all of the ideas that are around the podcast. It's kind of contextualizing what we explore in the show. It's setting the direction for future investigations that we do. It lets people ask questions that have come up for them, clarify things, meet other people that are interested in the same topics, and it's something that's really, really special.
Finally, it's probably your last chance to grab a ticket to our conference. If you have the time and will, please come out and hang out in Austin, April 7th and 8th, which is probably really soon, just depending on when we release this. And so I would love to see you there. You can grab a ticket up here and we'll get together. This is going to be a bunch of talks by some of our favorite speakers, workshops, hanging out, beer garden, eclipse watching. It's going to be epic. So I hope to see you there.
All right. And in the meantime, enjoy our conversation with Michael about water and the cosmos. We'll see you next time.
The scientific revolution starts now.
Podcast.
Let us see in the dark.
So you and I, we got to hang out, I guess it was last weekend, the weekend before. We were in Minnesota for this super huge conversion of nerds, like 15,000 math nerds in one city in Minneapolis. It was the American Physical Society conference. And so we talked a bit about all this stuff, and Anastasia wasn't there for that, sadly. Because I thought it was going to cost $1,000 to get her in, but when I showed up...
I was learning to rescue people with broken femurs from the woods. I was good.
I guess you're allowed to bring your wife, though, too. I didn't realize that until I got
All
there.
spouses, not just wives.
Sorry, all spouses, not just wives. Yeah, whatever sort of spouse you have. So, anyways, so we have to. So you know, your day job is you're essentially a. How would introduce yourself? Like, what do you do exactly? Because we have a lot of things we want to talk about. You're really interesting, dude. Your mind.
As far as I can tell, the conversation is water, consciousness, UFOs, and Western esotericism. Like, if this conversation unfolds in the ideal, those are going to be the 4 points. That
there's a lot to hit.
It's going to go down.
It's also totally fine, Michael. Like, if we don't get through all of that today, we can do another one of these. We don't have to hurry.
Yeah, I want to make sure that all of this is laid out for people who are not very well versed in it, because you have such depth in this, and so do you. You both have been studying water and a lot of the Western esotericism stuff for a while now. And so, like, I feel like my job here is to just slow everybody's roll. To just be like, hey, hold on. So where do you want to
So you were kind of introducing how you got into this, but maybe you can break it down a bit and just, introduce yourself as much as you feel comfortable to get people oriented to who you are, I guess.
I think that Anastasia laid out like the storyline that I like about this work, or like sharing, and it kind of starts as like currently I'm a post-doctoral fellow, like a studying cell biology, and so my career has been interested in math and science, and so like in high school having this kind of interest in like I love Star Trek, growing up watching Star Trek with my dad, so I was always introduced to this idea of like being interested in alien space, but also like camaraderie and fellowship in science for like science's sake, right? Like they have this really idealistic view of the world. I think I kind of inherited this, and so going off to like high school, I kind of shared my interest in consciousness, and so I feel like a lot of people are interested in like psychology, but for me, I was so like hard sciences. I wanted to study like biochemistry to understand like the physical basis of consciousness was my kind of interest, and so I go off to college at Colorado State University.
Okay, hold on one second. I got a question already. So is that just because the physical stuff seemed more reliable to you? Like the other stuff was just too wispy and ethereal? Yeah.
I didn't like, I mean, truly, I like had maybe even vanishing respect for that. Like, it wasn't like I was so raised in this like kind of, physicalist like mindset. And that's like what I like too, is just like, you can like prove this, right? We can like, it always seemed like if you were unanchored from like math and science, that it was hard to come down with like reality, to like agree on something. So I think it became this like language of which we could all like be sharing the same thing, which was like really hard for me to understand what's going on. And so just like, I think it just became really natural. Like the only thing that existed was objective physical reality, as like we kind of break down in the scientific method.
I think that's what's so attractive about math, is there is an answer. It was like the only class I remember in high school where there's like an absolute right or wrong. You could get into arguments even about the chemistry and the physics or whatever, but biology, it's all interpretation just at the end of the day. But there's something about math that's just really, I don't know, it's reassuring and relaxing, because you're like, okay, we can just calculate this. We all agree.
It's a known problem. There's an answer.
That's right.
There's a few things, and you never encounter the math that doesn't yet have answers when you're in high school. And so that makes a lot of sense.
That's right. And so that's where I was, right? I think that's like the culture in which I grew up in. Like, also wanted to adopt it for myself, right? Like, I wanted a firm basis to move around the world from, and science was that. So biochemistry seemed like that, like not to hang out or like be able to manipulate with consciousness, right? Of how I understand it. And so then I get to college, and I'm still interested in this. So I go to like the one biochemistry lab that studies neurons, and they're doing like actin and neurons. And so I like walk in, and the principal investigator is like very gracious in hearing this undergrad like describe their interest, and then is saying that there's no room there, but there's this new like PI, and so like walk out his door like down the hallway and turned it in to this new lab, the PhD lab, and there I studied yeast endocytosis, and so this was like not front and center for me at the time, but I was really fascinated by this. Like, what it was like taking this mechanism by which yeast took in stuff from the outside, endocytosed it in with the actin cytoskeleton. But this was like a really complicated physical mechanism of a cell getting all these parts and this protein machinery together to perform this like action, and trying to like piece it apart. So this is like wonderful training in a wonderful place with the perfect like, and the biochemistry class that I was taking at the time, I really like. Cell biology was fascinating because it's like uncovering this black box of the cell and how it works, but it was unsatisfying because, as opposed to like it was, you get glucose and you like get ATP out the other end, and that's a black box. And so it became structural biology, all the tools and the machines that were actually performing these chemical interaction or chemical reactions, that became so fascinating to me, because it went from kind of like ethereal, or again, this black box, to a physical mechanism. And that's so wonderful to be able to like gain control over these ideas.
Like, how does the cell physically, mechanically behave? Like, how is it actualizing its behavior? So it has some intention to like eat this chunk outside of itself, and it makes it happen structurally, and you can study the structural mechanism.
And when you get it down to something like yeast, it's actually simple enough to be able to diagram out, whereas inside of a mammal or higher order organisms, it becomes...
You got to deal with the brain. That's annoying.
With brains and hormones and circulatory systems and vasculature and all this like through cell transport. And so it just becomes really quickly an unyieldy problem. And biology can start to seem unhandleable because it's so vast.
Exactly. Yeah. Right. And I think you like touched on something that's super important here, is like the yeast was like approachable, and not only like experimentally because of a single cell, you can see it, and the genetics, you can quickly like iterate. But yeah, suddenly I'm not worried about the tissue environment, the cell type, the like where it is, and like there's all these things that it's like it's a nice like world to live and play in, because it's the appropriate size to gain comfort, right? I think that if I was just like straight into biology, it'd be like too vast.
It's interesting because like this is the principle of the model organism, right? You focus on something that's really small and manageable, but the weird thing is that we don't get really beyond model organisms because it's too hard to do any kind of science without them.
That's exactly it. And just like have a cool return with yeast later, but because yeast was like this perfect model organism to like transplant, and I began taking what I realized and was cool about this experience in undergrad was my interest in structural biology. So understanding the protein machinery, where each atom looks like and how it moves, is the field of structural biology usually. And so I started to like gain an interest in this. And Colorado State University is actually near like Prion research facilities where chronic, in Colorado there's chronic wasting disease with some of the local populations of like deer, elk. And so it's a study there.
Like
so
mad cow. For the layman,
For the layman, right. Biochemistry, maybe like many different names for these kind of groups of diseases where you eat some meat or nervous tissue, and you have a prion or a prion, depending on how you want to pronounce it, and it enters into your body and starts to like sequester other of your proteins to make this kind of infectious protein agent that then leads to neurodegeneration and these horrible diseases. And I actually, so because I was at Colorado State University, I got like a lot of background in the structure of this, how these protein forms are like mediated and transform and become these beta sheet structures, as we call them, or amyloid fibers. And so I then was looking to go to grad school, and my PI said, if you're interested in structural biology, you should go check out UCLA, where there's this person, Dr. Eisenberg, who's a guru in structural biology. And I like know nothing, but I do apply and go there, and then I interview with Dr. Eisenberg actually, and I know like all these answers to amyloid questions, and this lab happened to study the structure of amyloid fibers, and I like knew all the answers. And so I feel like I had the like best interview ever, totally on accident, because I just like walked in with like the background because it was like given to me. And what was like fascinating is how like yeast is such an important way of studying prions, because the first prions were really discovered in yeast, and it was discovered as this like mode of inheritance, of non-genetic like inheritance. So what yeast would ostensibly do is it have these proteins with these prion domains, and under times of stress, the prion domains will form these protein fibers in the yeast. And so this is kind of like precipitating out of solution, right? In the sense that this protein used to be bouncing around doing its job, but now it's sequestered into this like really stable protein glob. And this changes how the yeast behaves, because this protein is no longer like doing its job. And you can inherit this because it's so stable that when yeast buds, you can inherit like this blob, and it'll keep that in the daughter cell, this kind of similar behavior. And so this is a like interesting amyloid like tie back of like inheritance kind of, or how it's like used functionally in yeast. And so this model organism now becomes this thing where then how, if your daughter is also likely to inherit that same thing, how do you get out of this, right? How do you recognize it's not stressful anymore, like dissolve this aggregate, or how do you keep passing it on if you want it to go on? So they have these like enzymes and chaperones that will like break apart or not. And so after, like if you have a population after maybe 1 out of 100 yeast might like reconvert back, right? And so now when you have a model organism, yeast was really cool because if you're just studying one, you would maybe never understand the ecology of how you need a population of yeast passing on and stochastically entering and exiting. This is not the individual. It's like all of your model organism needs the community now to really observe this amyloid effect of inheritance and how it plays out ostensibly in the wild. And so I started to study like amyloid under this context where amyloid is this generic term for a protein fiber, and we typically study it in the context of Alzheimer's disease or Parkinson's or ALS. Something that's common between all these is that you have nervous tissue, and it's stressed at some point. Something happens, it seems, and proteins that were normally behaving one way start to form these amyloid fibers where they all stick together and they form a similar structure that we call a beta sheet. And what it is like if each one was kind of like a floppy strand, they all like stack on top of each other. They're like perfect hydrogen bonding the whole length of the fiber. And that could be like microns. So you could be like having millions of these proteins stacked up on top of each other.
And the way that you had this diagrammed out was basically just like a stack of pancakes. Like the proteins kind of flatten and then they just layer perfectly on top of each other. And it's just this like crystalline structure all the way down. And the cells can't actually handle them. And so they basically build up and get in the way of normal function. And this is
the amyloid cascade hypothesis, and largely what we would think about when like trying to interpret what's going wrong in these neurodegenerative ideas.
Is that still the main hypothesis for Alzheimer's, or is that
things are changing?
Yeah.
So I started grad school in 2012, and it's interesting because as we design like therapies to get rid of these fibers, and of course the idea of getting the structure of these fibers is then to be able to design molecules as therapies to dissolve them on some level, right, as motivation. And so when we get like something to recognize and clear these from our brain, the symptoms of Alzheimer's do not seem to go away. And this is actually like a big like thing in the FDA of like, we have this drug that was approved by the FDA that can seemingly reliably like get rid of or decrease these proteins, but like, does it change the progression? It's like maybe not reliable.
It was the Genentech drug that you're talking about, the Avacumab?
I think so. Probably. And if I'm now like 5 years out of really paying attention to this area since I've been like on the water journey. And so I know like 2012 to around like 2018, right? There's like this interesting shift where we're like, I entered in on this amyloid cascade hypothesis, but by the time I leave grad school, there's information saying that it's just, far stranger, right? Like it doesn't cover everything. There's having these protein fibers. But then that kind of brings up my graduate school work, and why it was like so curious and interesting of a journey for me was that I entered in kind of this idea of like these protein fibers are bad and we're trying to get rid of them. But the project that got put on actually happened to be on this protein called FUS, which was a known ALS-related amyloid protein. But what I found was when I kept getting structures of this protein from its like prion-like region, it didn't have the normal amyloid structure that we see. Normally we see like when you're talking about that pancake, that flat structure, you see these like long, like interactive faces a lot of times between itself. And this makes for a really stable structure. But the ones I kept solving were kinked. They have these sharp bends in them. And what that means is it's hard to make a flat interface, right? That if you're like kinked at a right angle, you can never like bind as strongly. Right. And so it seemed to be this like mechanism that was built into the sequence of this protein FUS, where the structures it would form wouldn't be as stable as the traditional amyloid that we would normally like come across in disease. And this entered in with like a series of other ideas at the time, where it kind of like maybe was this interesting building block of maybe there's like some in-between state of, diffusing happily in solution versus like amyloid fiber. And there's maybe like a metastable thing where you can get like a medium stable or size amyloid fiber. And this would actually be helpful in gene regulation because these are usually RNA binding proteins. So if you have DNA being turned into RNA being turned into proteins, as like the information transfer, these proteins are binding RNA, and they're really important for regulating how those RNAs get turned into proteins later. Right. And so you can imagine by having like a weak ability to like stick together and bind on RNAs, that you can have a lot of control over the system. It gives the cell a lot of control. And this is
like a really not super smart way, right? Like it's not like you have to have a centralized brain. It's literally that you have taken a structural approach to the control of available reagents, which is
Like
really
feedback
cool.
Yeah.
loops and so forth.
Feedback,
like negative
right.
feedback, I
but I'm
guess.
also
And it's like
like,
crowdsourcing, like the local environment.
yeah, exactly. Where it's like, okay, so you have this stuff that works when conditions are right, and when conditions change, it starts to agglomerate. And I just see this in my mind's eye. I just see it like a local region of the cell where a lot of it is agglomerated, and it's like a mixture of protein and RNAs. And
Yes.
so, as the new RNAs are being manufactured, they're being recruited to this globule to prevent them from being made into new proteins. And then, as conditions start to go in the other direction, those interactions lessen, and all of the sudden, but it's such a tunable feedback, because if it's electrochemical, if it's pH based, if it's responding actively to the conditions of the cell, you have something that minutely will start to dissociate or reassociate in a way that basically is immediately responsive to the conditions of the cell. That's really cool.
That's exactly it. And this is the beauty of what like my project turned into, was exactly what you said, of like kind of starting off with like what's going wrong into disease, was now like this uncovering of how these proteins were in this organization that you talked about. And the one that you're specifically referencing becomes something we call a stress granule, of that when the cell is stressed, there's these conditions form where these RNAs and proteins, as opposed to going and like being turned into like turning the RNAs into proteins normally, they now end up in this stress granule, and we might call it like a coacervate or a condensate or a membraneless organelle. And what membraneless organelle is implying is that this is a organelle or a like reaction hub for the cell, kind of like the mitochondria might be, or the nucleus or the Golgi might be. It's doing something for the cell. It's organizing compacting components to do that. But as opposed to wrapping it in a membrane, now you're like continuous with the aqueous like solvent solution, right? There's water inside, water outside. There's no real like barrier in between. And so this is now like became a revolutionary way to understand organization of the cell in biology and biochemistry and cell biology that started to come out right like 2015. Is that as opposed to wrapping something in a membrane or just diffusing freely, you have these microdomains, which are these granules, these like coacervates or condensates, that seem to concentrate particular RNAs or particular proteins or do particular reactions. And so this, as opposed to like,
So when you say that they concentrate particular ones, was it possible to like look into the cell and find that various proteins or RNAs were clustering in different areas of the cell? Or were they
Yes.
were each one of these organizational clusters a mixture of multiple kinds? Like,
Yeah,
are they self-selecting for purity in the sense?
Yeah, to different degrees, yes. And the thing is that the biggest give or tell of what kind of an RNA gets sequestered into one of these granules is like how long you are, right? Like a long non-coding RNA may even like structure this. So in this sense, yes, specific things, but in a sense, it's like very general and very like loosely selective. So like when I have a plate of cells under the microscope, and I like am trying to stress them and study these granules in a stress condition, like 10% of the protein I'm looking at might go into these granules. They're really bright because it's like really concentrated. But remember, it's like only 10, but the rest of it's still diffusing freely. And so, dramatic effects, but we're like a population size, right? It's not like you just like hang out here, you're stuck here now forever, except when amyloid fibers form, right? Now you've entered this super stable structure that becomes irreversible and it's hard to get out. And so that's this big difference, was this idea of like the amyloid cascade wasn't working very well. But my postdoc actually started on this concept of when you start to concentrate these things and you form not a solid, but like a liquid, a continuous, but a separate liquid phase with this concentrated protein, eventually the idea was that amyloid fibers would start to grow from that concentrated protein solution. And this would lead to the amyloid fibers in these diseases, was that under the stressful state, you got all these proteins together, and then they're just going to randomly nucleate into and grow amyloid fibers.
But is it the stress that's the source of the disease then? And these are sort of like the ambulances that show that like this is just a downstream symptom of what something else that's dysregulated in the first place?
That's what this story like tends to turn into, right? And this becomes a confusing thing of now it's trying to like pick up cell biology to turn this structure work I did into something useful for understanding what's going on in the cell and biology. And where like the hang up becomes, because right, so I can get fibers out of it, and it doesn't necessarily like change the health, right? Like, then what am I looking at? If what is like, it just became too vast of a problem. Like other people, there's many ways to trigger stress granules, right? If I like uh, make it too hot, make it too cold, make it too like anything that's not normal, right? You're gonna like get a stress granule, and like they might have different compositions. It won't all be the same. This is interesting because different stresses, you get your selection for different pieces. There's so many ways this story can go. So many. Like the decision space by which to like manufacture hypotheses is vast and like deep. Like I can say anything and like it sound pretty good, like be worthwhile, right? But because I couldn't,
Well,
right?
kind of, right? Because it's like the problem with it is that if you're looking at a symptom and you're saying the symptom is the cause, then whatever plausibly good mechanisms you're proposing are not fundamentally dealing with, well, hold on a second, it's the stress in the first place that's causing it. And you can go around putting out fires all day long as much as you want, but you're never
Stress
actually
is
going to
just
put
such
the
a
fire out.
sufficiently ambiguous concept, though.
That's
Right.
true too. Like all of this, right? It just becomes really nebulous as like where to go with this idea. Like, what is stress? Is it something like what is stress to a cell in a dish compared to in your body, right? Have we ever observed like I went through grad school wondering the whole time, has anyone ever seen a stress granule in tissue, like in a person, right? Or is it
Did
just
you
a
ever find
product
out
of
the
like,
answer to that question?
oh, it's probably, yeah. The answer is these things definitely show up in
Okay.
bodies somewhere at some point in time, almost assuredly. I
But no,
think
has
like
anybody actually like done like a thin section and like found it?
I stopped looking for that information. I think that you would like find something like that these days. I'm pretty sure something like that exists, but I haven't actually like found it and seen it. I don't
That's
really
fair.
doubt, because stress granules in particular are a unique like thing that's like worth that debate. But there's many versions of these like condensates, and one might be the nucleolus, which is where in the nucleus you make the rRNA for the ribosome, which is the protein machine. The machine that turns RNA into proteins is the ribosome. And so there's a particular area called the nucleolus where you manufacture ribosomes. And this is a common version. You're going to find these in all cells, right? And especially now when you're talking about like growth or cancer, things like this, now suddenly becomes really interesting to target these kinds of loosely organized granules for their like effects in machinery, right? It gives us like this new tool or this new way to interact with trying to find medicines or counteract disease. To appreciate this, so stress granules is like one version, but there's others that we know for sure exist all the time, all over the place, very essential. Stress granules are one that's a little bit more.
I mean, I'm looking at it
Yeah.
looks like they do at this point have thin sections for
Okay, that
for
sounds
stress
right.
granules.
Yeah,
I mean, it seems to me
Which is
like
pretty cool.
living is stressful. And
right.
To varying degrees. That's why they say you should
Yeah.
meditate.
right, and I feel like the older you get, probably the more accumulation. Like I feel like being stressed in all these situations is somehow damaging, and there's some sort of healing that goes on. But inevitably, since
Yeah.
we don't live forever, we don't possess an infinite ability to heal, and it must just accumulate. Like it just seems like these degenerative. I mean, look, early onset Alzheimer's, like that's an interesting puzzle, but like people being really old and their bodies falling apart kind of makes natural sense to me in some sense.
Everything decays.
Right.
Yeah.
Yeah.
And so stress is just like this inability to respond to change that accumulates over time, then so it goes.
Well, the body's
It's so
supposed to be
gross.
able to have mechanisms by which to clear that, right? So it's like I know that there is. I can't think of the
like
pathway right
on
now.
like on like an individual level, right? Like you can deal with one
Well,
problem, but then you get another
yeah.
problem and another problem and another problem.
Well, and that's the thing, right? Is like I think that the longevity research should be focused on getting people back to a baseline where the problems don't continuously add up, right? Because I know that like the lymphatic system is the brain's lymphatic system, and so when you get enough sleep and you're properly hydrated, what it should be doing is it actually like washes across the brain as you sleep, and so it's getting rid of all this misfolded stuff that functional cells should be getting out. And my intuition is that you start to accumulate problems where you live a life that's not possible to do. Whether it's
Yeah,
because you're not
and
exercising or you're not sleeping or you don't have the right micronutrients or you have circulation problems, or you're a smoker, or you're on hormones, or you are exposed to glyphosate. Like, there's all these things that add up over the course of a life that prevent
Yeah.
these things from working. And when they've stopped working, then you start to see the accumulation of damage that over the course of a life creates all of these problems.
That's exactly like, and the origin of this idea, like the amyloid cascade hypothesis, is kind of building on that. What like Shiloh was getting at is that like once you start with some of the stress, it just like piles up. You can't get rid of it, and right, and we do have these built-in mechanisms that you're getting at, right? Like when we sleep, we like flush our brain, seemingly so important. People with Alzheimer's like interrupted sleep all over the place, right? Like all these things at some level, and I'm harping on this because I think this is what kind of like the water story becomes is a different like way of view of like there are these natural cycles to how health is like carried out, and we always have this like cleansing thing. And so I mean even like some I'm not super expert on this, but like cancer research, like many like things that might be tumors starting in your body, right? But what is the difference between becoming one or like your body dealing with it appropriately and disappearing, right? Like when you start to treat cancer versus like when something that you can like handle. And so this appreciation of how our body has built in all of these ways to erase away or like deal with the stress that we've gone through. But there seems to be something that happens where you start to accumulate, and it feeds forward in a really destructive way. And how we get out of that, and there's a few ways, right? Like at the end of the day, like the idea essentially is to like make a drug that counteracts that horrible like feed forward cycle. But there's also this other really important part to me that I love focusing on is like what are these cycles that we're naturally doing that prevent us from that, right? And so this is kind of the thing of like entering under like Alzheimer's disease and trying to prevent amyloid, and then being kind of switched to like, oh, what's going right naturally first is like what I want to know now. How do we like keep these cycles going? And I think that's also where like people describe Gilbert's Ling work as so interesting, as really trying to like put like his big thing he puts forward is this like cell has a resting state and an action state, and it's like cycling between these two.
For anybody who doesn't know Gilbert Ling, can you give like an overview a little bit?
Yeah, and maybe it's like too premature to go into or like come up later, but what I encountered in then trying to understand how to turn the amyloid into like my structural biology background and understand this amyloid disease into something useful like in cell biology that we can like turn into a drug later or help out with these diseases. And I was just really struggling with the cell biology. I just started this postdoc in cell biology, and because in structural biology it's so like a mechanism, it's like rigorously worked out in a really small system, like a protein. Like a protein moves some atoms around, and that's your chemical reaction. It's like a really nice story, but you can't do this for cell biology. It's just like too vast, too big, as we kind of alluded to earlier.
That's why cell biology is almost always pathways rather than proteins, right? Like biochemistry is like we have an active site inside of a protein. This is the way the chemical fits into the active site. This is the process by which it reacts and like what residues are involved. And then when you get to cell biology, it's very much like, okay, so we have this class of proteins, and this class of proteins responds to this stimulus by being modified in this way, which affects their activity as a whole. And these are the cascading effects that are happening.
It's almost
inside
like
of the
these
system.
information networking projects.
Definitely.
These chains of
feedbacks
they look like
and
nodal
loops.
networks.
Yeah,
They are.
absolutely.
And that's what it turns into, right? Of like these pathways of like genes or proteins talking to each other. And so as opposed to like structural biology, where my mechanism is like moving atoms through 3D space, now it's like moving information or concepts around the cell, right? Like if you have a signal from the outside, I have to move that concept inside. And that's like, there's different mechanisms, like might be phosphorylation, we have different chemical mechanisms, but really, right, like I'm putting out like a black box or a catch-all for this. And so
and like somebody else will go through and do the protein biochemistry to figure out if your mechanism is reasonable down the line.
Or that might be like part of it, right? Yeah, of your paper, right? But yeah, every scientist is trying to like answer a question that they're trying to answer, and they have their own tools, and you can't be everything at once. So it makes so much sense that cell biology, you can't go in and like study all those like proteins at the right level that you want to, but you do see that they're talking to each other, right? We see this, like they'll go hang out nearby each other. Some motion happens, right? So this is really liable. But the issue is like making big grand stories from this of like, how do we go from like detail of like, okay, so then this issue is like, I think that like the central dogma as it's taught in high school is this thing that gets around biology that I have no idea where it came from, but it's like DNA to RNA to proteins, how information flows or
Francis
something like
Crick.
that. Right.
It's like it's in his
And
notes. He laid it out like after they had discovered DNA, and he was basically like, "This is the direction that things go." And his point was that it's the idea of the central dogma has actually been warped a little bit because
yeah,
everybody treats it as being kind of this outdated thing. But his point was that you could probably go back and forth between DNA and RNA, but you could never go backwards from a protein. He was like, by the
okay, that's
time that
interesting.
you've
That's his
solidified
intention.
information inside of a protein, it is unidirectional. Like you can't go back and
Right.
modify nucleic acids on the basis of the
Okay.
protein.
All right, fascinating. That's nice to know his like original intention because I feel like how it gets realized is like confusing to me, where it just becomes kind of this like guiding way of how like things are organized. But then as you go deeper into biology, you just keep uncovering, well, the RNA can be modified in all these ways to either like help it turn into protein or not help it turn into protein. You can do all these different like signaling things of like degrading mRNA so it doesn't get there. There's all these ways to modify that pathway. And so
And not to
then
mention
when
the
you're trying to
proteins are modifying the
modify
DNA,
too,
like or
right?
transcription or whatever.
They are, but
Yeah,
the
it comes
idea is that his point was not that they can't interact with the DNA. His point was that they can't like change the DNA. Like by
right.
the time that you get to a protein, the protein does not have of its own volition instructions to go in and start modifying what the DNA says. Like the DNA can modify what the
Yeah.
protein says, but the protein can't modify what the DNA says.
Is that true?
I mean, intentionally, I don't think that there's any proteins whose job it is to go in and modify DNA.
What about those ones that results in recombining and so forth?
But those are supposed to be repair proteins. Like you're talking about like double strand breaks or something? Or?
Yeah, I guess so. Transposon type
Well,
interactions?
transposons are nucleic acid sections.
And there's no proteins
There's
involved?
usually no proteins involved. There's just like there's like repeats on either side, and those repeats activate a protein
Isn't it a protein that cuts it?
It is, but
I don't know. This is
Yeah,
no, yeah,
doesn't
we're
matter.
in the
Even
weeds.
if we
We're in
get
the weeds.
Yeah, and if you get like abstract enough, like protein is always carrying through the DNA, so the DNA doesn't get to like just sit there, right? It's maybe the queen ant, but you don't get to like just sit there without all your minions carrying you through the ages. And so at some level, the proteins are talking to the DNA. They're like in an ecosystem together. But you're right, like that information flow, that sounds like it's interesting to hear the original like intention of this, because I think that grounds it much more than like how it was taught to me in high school. But this idea is that like all these like pathways get to it's so vast, so big. Like, all right, so now if I want to say one gene, how is it like modified on the RNA level? Is it like upregulated or like cut up under these like stress conditions? Is it modified after the RNA level? Like when the protein's there, do the stress conditions change the modification of the protein? How does that change how then the whole thing like repeats, right? And so there's so many details that I was faced with. I got kind of like frustrated, like overwhelmed. And this is actually right around because I didn't know where to like go. I could go any of them, and they all turned to postdoc projects. But I just know this one like detail. I was really like trying to figure out how like if I want to know what any of one of those things like did, how could I predict that, not just one at a time?
You wanted a larger
so,
rule, which is like the thing
yeah,
that biology and most of science moves forward by. You have a system that seems
right.
individually very complicated. And
Yes.
instead of having to discover how each and every single one of those individual systems works, the ideal would be to go forth and be able to say, this is the rule for how systems of this type operate.
Right.
And now we can
Yes.
manipulate it and control it and affect it and change it because we understand the rules by which it operates. Which is
That's
like a
right.
better approach, I think, in general to science.
Hey folks, quick interruption. This program is entirely supported by our listeners. That means we have no ads, we have no sponsorships, we have no products we're hawking at you. We're just supported by people who care about these investigations. If you have the tiniest amount of money to break off from your paycheck and support this podcast, it matters a lot. It's what allows us to spend more and more and more time bringing you better and better investigations. So consider giving just a couple dollars a month over at patreon.com/demystifysci, and I will be eternally grateful.
Because we have so much basic research already done.
So much basic research already
And
done.
it's
And that
just-
was
And it's
I was
not fit
reading-
together.
Right. And I was reading, I was going through all these papers. It's like, why should I mean, how can I like start a paper? I haven't even like caught up to the material yet.
Yeah, I know. Like,
Right.
good luck
Like, how
catching
can
up
I,
to the
like-
material.
Yeah, and that was it. It was just like, I mean, this field was growing so big. It was just like there was no way that I could possibly like be contemporary with choosing a project. And so this was kind of getting to this point where it's like I just started my postdoc, and now like COVID is hitting, the pandemic is happening, it's like shutting down, and I'm getting a little bit like frustrated because I can't like go to work. I'm like stuck like thinking a lot at home, and I'm kind of like wondering, well, this is hard. Like it's hard. Like I could revisit anything I want for my postdoc, and this around this like nexus is actually where I started to repick back up like the UFO and alien like interest I had from earlier. And it goes
That's a
along with
jump. That's a leap.
that is a jump. But where it comes from is you were talking about I was searching for this bigger idea to hold all of this like niches of biology. And so what these are called are theories of everything. And what ended up happening was I started watching this theories of everything YouTube channel, right? Where again, like a peer appears, I assume of I started to listen to these like UFO and alien like government whistleblowers on the theories of everything channel. And that actually was really interesting to me. But most importantly, it led me to Michael Levin's work, right? Because Michael Levin, as someone you talked to, is also that. And so imagine this like fascination when I came across this like mind who now interact with biology in exactly as you're describing, like what are these master control things, right? When we're zooming out, what are these like bigger things that we can manipulate to have a large scale view of what's happening at that detail, right? Like we don't need to understand all the how you modify RNA and turn it into protein. We don't need to understand all the modifications if we have like a bigger rule set to zoom out and like have some control. And that's what I was looking for, right? Because like going from the protein up to the cell, up to this human body for amyloid at each stage gets so vast in how you can like go. And so this became what I was looking for, is these like ideas that started to like really like condense and bin like concepts together. What are like the really important like knobs to turn to get something your system to do something that you want. And Michael Levin's, of course, puts together as like bioelectricity. And I think that's just like fascinating. I loved like hearing him talk and how he described what he was looking for. And it changed what I did, because then I like was really interested in looking for like how would bioelectricity be affected by the stress system, right? How do I take what like Michael Levin's been doing and like try and apply it in my own research as this like master control system? But then like changing what I'm like reading. And so this is where I started to come across Gilbert Ling, because in like these like alternate ideas of cell biology that have been passed down. And so in looking for theories of cell biology that encompass like a lot of space, have a lot of predictive power, I would keep I should actually start up with how I came across like Gilbert Ling in the first place. Was from my like background in amyloid structure and trying to understand what's going on in cell. I was trying to understand what causes like a protein to go from just hanging out in like a test tube or in the cytoplasm of a cell to being in one of these granules, these condensates, either in a test tube or in the cell. And so what I became convinced was that like I knew what the proteins were doing. I knew a lot of the physics of protein interactions, technically an expert in that, but it was the water. Like that's the only place it could be hiding, right? That's what was missing. And so what I grew up is like, if you have a hydrophobic part of a protein, meaning that it doesn't like to form bonds with water, it doesn't hydrogen bond well with it, then water forms like a clathrate around, like a cage around it to like kind of like seal it off. And so water will like optimize bonding with itself around the hydrophobic. But that seems like that's what would happen around the hydrophilic. Like if you could form a hydrogen bond with water, you would just see a cage around you. You would also get a cage. So these are the like details. I realized I had these blind spots, like where these energetics are and how it's working. I was in the water shell around it and what the water was doing in these interactions. And so I started to read about this and the association with phase separation, this field I was in, of how you go from like being in solution normally to in this concentrated, like phase separated kind of condensate.
And so
I had
for
this,
people to lay this out a little bit, it's basically the idea that water, when it is in bulk solution, is just kind of these molecules that are bumping around, forming temporary hydrogen bonds. But at surfaces, the water is able to form much more permanent structures, where it's this crystalline shell that interacts both with itself and with whatever object it is solvating. And so you were like, there's something about the way that the water must form those shells around the object, which is the thing that is the master regulator.
That's exactly it, right? And so in the basic sense of looking at like the formation of an amyloid fiber, right? Of like when you're going around freely in solution to then being stuck permanently attached to this like protein fiber blob, or even an enzyme like forming a dimer, is that for these proteins to come stick to each other, like first of all, they have to find each other, and that is going to be mediated not by the proteins, but by the cloud of water around them, right? You're not feeling the protein, you're feeling the water cloud. And the second thing is that when you get rid of that water cloud, that's when you're precipitated. As soon as you get rid of your water shell is now when you're the amyloid fiber, right? And so when you have an interaction or like that phase separated thing where you're going from like hanging out freely in water into now being in this like dense granule with like every this liquid concentrated protein phase, the proteins tend to form more interactions with each other. We might call this as like stickers hanging out with each other. And these stickers might be between like spacers within the protein. And so even for two stickers to like get together and form a more stable interaction, you have to get rid of the water between you two to like have that protein interaction, right? And so then in the test tube, where we have like this bulk water, it's like very, very free. All this water is like interacting. But then as you get close to the surface of a protein or even the surface of a test tube, then your behavior changes because that surface changes how you can freely move. And so whereas like in bulk water, hydrogen bond might form and break in like a picosecond time scale. So yeah, like, yeah, seconds, like milliseconds, microseconds, nanoseconds, picoseconds. Picoseconds are where you're like forming and breaking hydrogen bonds, but then water next to a protein might have like a 12 picosecond like lifetime, or it's like or 2 picosecond. It could be a longer hydrogen bond lifetime because this huge protein object like makes them more stable. It's not moving around, right? You don't have the degrees of freedom. If you have two tiny water molecules, it's really hard to find each other tons of degrees of freedom. But if you have a huge protein molecule, then you've really like refined or decrease where water can diffuse in that local area.
That's still a pretty short bond lifetime, though.
It's still very short. And this is actually kind of interesting. Yeah, even water and like really like dense gels is still like moving constantly. I think that Shiloh, when you're looking at like in Shiloh's research with water, like these hydration solids, right? Like when you're looking at AFM and you're like bouncing a needle, and there's this like if you vibrate slow enough, it deforms, right? But if it's too fast, you like can't move it, right? And so it describes this like blocking or jamming of water molecules can't move past each other on a time scale. And yeah, so like when you start to get like water molecules like buried in a protein, buried deep, and so like all their 4 hydrogen bonds are satisfied by macromolecules around them, now you're getting into like millisecond and microsecond time scales for like changing a water partners out, in or out, right? Because it's so stable. But
I did want to like comment on something earlier, which is just that these hydration structures, the individual water molecules can swap out and the structure remains. So there's like when you said that they're long, I almost interrupted you, Nasi, when you said that they were long lasting structures, because it's kind of true, but it's not this. It's more of like what is it, the ship of Theseus? It's kind of that situation, right, where you can swap out the individual planks, but the ship still remains, and right. There's some of that going on as well. So despite all of these kinetic that's why it's so difficult to study as well, because the traditional means that you would use to study the motion of the waters won't necessarily tell you about the structure itself, because it persists despite that.
So it's like the larger organization that persists for a long time. I imagine that the hydration shell around the protein is basically a permanent structure, barring severe changes to the environment.
But it's always got different players involved. It's like a stadium. Yeah. It's just people are leaving and filtering in and out.
And it's like, it's a question of like, how much do you need to study the individual molecules coming in and out versus simply the appearance of the layer, period. Right?
Can I ask you a question? This is... Is there a sense of these hydration structures? Do they have a permanent sort of lattice structure, or does it depend on the protein that they're hydrating?
I think it depends on, so very much depends on the protein. Like everyone, it's going to be your protein's fingerprint.
Okay, but will the waters arrange in a like hexagonal fashion, or will they arrange? Will they always arrange in different? Because there's only so many ways you can pack waters together, I guess.
This is fascinating. So this is where like I don't have a good answer for you, but I have what I think is like probably what's going on, and the answer is it seems as though there's kind of like an infinite continuum of like possible ice structures, and
Okay.
like ice has like 17 structures, or
Right.
like so many that we know about, or not even like ice structures, but maybe like glassy water. So there's a few things to like discern here. Is like there's a liquid or like a gas where you like are just bouncing around. And the biggest thing about gas is like you don't have a neighbor for a long
Yeah.
time. Right. And then when you're a liquid, you're like always having a neighbor, but a lot of freedom. And then when you're a solid, technically you're crystalline. And so technically a solid, how I'm using it is you can like move a brick anywhere in a wall and like rebuild the wall. And in this sense, an amyloid fiber is a one-dimensional crystal because you can just stack proteins in 1D. But then um
And the hydration layers kind of have to be some sort of crystalline form, I guess.
so
It's,
this is it.
I don't know. That's the
Yes.
question, I guess.
Yeah, this is, and so my expectation is that hydration layer, so you can sample many different kinds of ice or acceptable waters, and those will have different densities. And we'll get like more to this later of like what's allowed. But yeah, what uh I would expect is going on. I think that this really good is that what's crazy is that it doesn't seem like there's much energy difference of like kilocalories per mole between like the different ice structures, not like a ton. The issue is that you have to pay for the whole ice cube to transition, right? But on a small local area, one hydrogen bond is enough to kind of form a local like ice structure, right? And so in this sense, you can think of the hydration coat as like an amalgamation of all these kind of tiny like metastable ice structures, and some of these are compatible with each other and move around. And the thing about like different water crystal structures and ice structures that they have different, I mean, densities, but also behaviors. Like, are the protons like organized in certain ways or not? And so now you can imagine like forming stiffer or less stiff structures. And so you may not have the same ice structure, right? It may be a very, very small of the possible ice structures that you're sampling in that one area. And that's what gives it its like preferred environment. And that's going to have a general behavior compared to somewhere else. So like ice binding proteins, the greatest place to study, because you can have antifreeze proteins in things like the snow flea or like these bugs that live on ice, or like deep sea fish, kinds of things like this. And so if you form an ice crystal that's too big in your cell, it'll puncture a membrane or the organelles and hurt the organism. But what they'll make is these proteins that bind the ice surface, and then they prevent the ice crystal from growing larger. And so now you have a protein that will inherently recognize water that looks like ice, which is that hexagonal lattice packing, right? So you have an organization of amino acids that always makes the water organize in a way that looks kind of like ice. So it'll orient to the ice structure. And then you have the opposite side packing the amino acids in a way where it really fights the ice structure. So that is not compatible, right? Ice crystal structure is not compatible with the opposite surface of an antifreeze binding protein. And this is then going to
I
be
mean,
like
the
what really
reason I
fights it.
asked, like, this is a little bit off the beaten path, but I'm really interested in Pierre Marie's idea about the oceans potentially being a source of microwave radiation. And the requirement for that is obviously that there's a transient hydrogen bonding network that produces a lattice, just like any other black body, right?
Yes.
Like, and so I'm always trying to push him to understand ways of understanding that lattice. Like, if it's a hypothetical lattice, obviously in the bulk ocean, but the ocean's full of
Right.
solutes and full of all sorts of biomolecules, and there must be all sorts of different hydration structures just all throughout it.
And like weird stuff
Yes.
definitely happens at surfaces. Like I know that this is a
But we usually think of surfaces like the bottom of the ocean or something, but I'm like thinking there's surfaces all throughout the water as well.
Right. Like a solute has a surface.
Right.
Yeah. So all surfaces change the water. And so there's water, there's like bulk water, pH 7, room temperature, like there's pure water, which never exists. It's always like auto hydrolyzed. And the big thing is like dissolved gases, like anything you do to water, shining light on it changes it. And so like, but pure water with nothing else in it is its own thing. And every time you put something in water, it changes that like structure's behavior. And this is like the big reveal from what I was reading about all this like chemistry, and like where Gilbert Ling gets in is like, and how you like abuse or get to like will use like trying to cycle between these like water structures. So it comes in, but this is fascinating because it's so useful for organizing, because now you have these like different kinds of water organizations, and some like each other and some don't. Just like the ice binding protein, one half likes ice and the other half despises it. right? And so now you're organizing your entire cell by how your affinity for water is, right? And so this is how that surface changes the water structure is going to be how you're going to be then like pushed by the water to organize, right? And so it's going to be almost this desire of like pH 7 bulk water to like return to itself or to like not be attracted somewhere else, right? You're like playing with these concepts.
I think this is at the heart of the paradigm shift that we're circling around to, is that we tend to think of biomolecules, or let's say like the biological material, as having organized the organism. But it seems like water is actually giving a lot of the mechanical properties to the organism at the end of the day. I think this is what Özgür was like dancing around about and getting super like, hyped up about at the conference too, is he's like,
He's quite right.
This idea that the structural integrity and the action of a lot of these cells, these cellular entities, let's say just biology, a lot of it could come down to the water doing stuff. Like the water is the actor, actually.
He actually, when I talked to him, had this really interesting take on it where he was like, in the most extreme version of the theory, the structures of terrestrial organisms are intended to carry a specific type of structured water with them as they leave an aqueous environment. And he was like, we have it backwards because we think of the proteins and the membranes as being the thing that is the cause, but really the water is the cause, and the proteins and the membranes are the necessary infrastructure to maintain the cause once you start to dry out.
Right. And I tend to think of this as similar to like almost Michael Levin's concepts of like what are you like driving? And in the sense is like the DNA is not like driving the cell. It's kind of like driving, it makes the machinery that the cell can use to like respond to its environment. And so I think in this way is like we're really trying to learn like what all of the like molecules are kind of like, they're having this conversation with the water. They're kind of in a sense like driving it and structuring it, but they're also like building the water structure by which they can then receive this like information back to and like do with this. And so I think it is like really understand this like high level like computation of like how water is like the space-time medium by which like we're communicating information in the cell, where if it's like gravity in space or whatever, then distorting space-time is like distortions in water is information transfer in biology. And like water actually like allows for very like fantastic versions of this, because you mentioned Pierre Marie's interest in the microwave background and how like water can do this. And I think this was like fascinating to me, is that the essence of why water is like so special is that if you have is this like desire to be tetrahedral when it like also wants to collapse and be closer. And so if you have perfect spherical packing, and so you have one sphere and you're like surrounded on all spheres like as ideal as you can get, you have 12 neighbors, like 6 around you in a hexagon, 3 on top and 3 on bottom. Right. And this is actually kind of like the crystal structure of hydrogen sulfide. So hydrogen sulfide is about as close as you can get to water, as opposed to oxygen. You just go one down, like the group to sulfur. And so H2S is kind of similar to H2O, but it doesn't have as strong of a hydrogen bond. So the crystal structure, from my understanding, is actually kind of close sphere packing, right? And when you're doing close sphere packing, you're optimizing for basically van der Waals forces, which is kind of so we're saying like there's an attraction when two surfaces are touching because of instantaneous fluctuations of their electrons, and this is what we call van der Waals forces, and it's kind of how oil like hangs out together, right? These are like oil molecules sliding over each other, stay together by van der Waals forces, and this leads to this like close sphere packing in hydrogen sulfide,
and it's
where you have
interesting
12
because
neighbors.
hydrogen sulfide is, I think, the worst of the acids for organic materials. Like hydrogen sulfide will burn you from the inside, right? Or is that?
Interesting. Or is that sulfuric acid? Or am I missing? Because hydrogen sulfide, is that one dangerous to humans? I
Am I
don't
thinking
actually know.
fluoride? Hold on one second. Maybe I'm totally wrong.
Yeah, so I know that for hydrofluoric acid can do some odd things too. Like that's a weird one. It's not like a strong acid, but it's like hyper strong in the right way.
Yeah, I think hydrogen fluoride is the worst one, if I remember correctly from high school chemistry.
It's got some really like unfortunate consequences. but not a strong acid technically, which is like
You're
what's
right. You're
fascinating
right. You're
about
right.
it.
I think it's hydrogen fluoride. My bad.
All right,
Yeah,
wait. So why are we talking about hydrogen sulfide? Sorry.
so hydrogen sulfide is talking because it's one of the closest similarity molecules to water of like the right size and shape. But in the crystal structure, it has spherical packing with like 12 neighbors, but water has tetrahedral packing with 4 neighbors. And this is like fascinating. And so what this is saying is that water for the ideal packing like pushes neighbors away farther than they want to be naturally.
Mm.
And this is why an ice cube floats, right? And this is because the hydrogen bond is so strong that it's more powerful to have 4 hydrogen bonds than 12, plus 2 fingers near neighbors with van der Waals in water,
Gotcha.
right?
Gotcha. And so you're saying
so,
that results in this sheet, this hexagonal sheet appearing as well in the middle?
as in like the structure of ice, or
Yeah, or I guess in the sense of tetrahedral packing. So
yeah. And so water is always in this fight of like wanting to be tetrahedral, and when you cool the temperature enough and you strengthen the hydrogen bond relative to like thermal motion, you get ice cube. But the thing about the ice cube is that it's like drawing a vacuum in, like because you're farther away than your nearest neighbors would want to be to optimize van der Waals. This is why ice floats, and so it has this like odd like vacuum. And so this is what makes water so unique. Is like water is the most dense at like 4 degrees Celsius at our pressure, and then every degree you cooler, it gets like more volume. So this is absurd because like if you take like almost like any metal and you cool it, it shrinks down as it forms a solid, right, metal. But now with water, as you like cool it, you expand, right? And so that like vacuum is like kind of where that's going on some level, and that's like fascinating. And so this means in liquid water, ice cubes flow is like a density of like 0.9, right? Grams per milliliter, but then water is density of like 1, right? And so it is more compact than this. So you're not always in that expanded form. A lot of water is in this compacted form. And so is there an infinite range of like forms you can be compacted in? And this is interesting because then like when you take ice and you like either start to put pressure a lot on it, you can collapse it down to a much denser form. But this isn't density 1 like liquid water. This is density like 1.2 or 1.14, right? And so when you then collapse ice's structure down, you skip water's density and go straight to a collapsed structure, right? And so this is like the true stuff again, like where Gilbert Ling is starting to like really abuse water's like potential. Is now you have this thing where like it seems like you can almost store energy in this like organized entropy, like low entropy tetrahedral kind of lattice structure. And so, but then I guess like as you're getting cooler, you should be like losing and getting closer together. But instead, you can like pack it, like keep yourself like farther apart than almost any other molecule in existence.
So
And
can you introduce this guy to us?
yeah. So
Ling.
now it's like two-phase water is this theory of called two-phase water, where liquid water is best described as a mixture of 2 phases, a dense phase and an expanded phase, and they're always like rapidly interconverting. And this is an idea that I think is best put forward by Dr. Philippa Wiggins from New Zealand, who's like passed now. But I remember reading a paper like right when I was going through this whole like water train, being like, this is absurd and insane. And then 2 months later, coming back and being like, I feel like this person was inspired. Like I think that she truly like saw like beyond, and it took me a long time to get there. And she describes this like wild behavior, but she heavily cites like Gilbert Ling of like different salts that will like enforce or break down one of these 2 like states of water. And she cites a lot from Gilbert Ling, and so Gilbert Ling preceded her. And so this is a good time to like get into what he talks about. And so Gilbert Ling is a Chinese American biochemist who came to America in like the 1940s, and he was a biologist, and he was working. He basically came through this like exchange program with China, where the like a physicist, a chemist, and a biologist came over. The physicist and the chemist went on to win Nobel prizes, and Gilbert Ling like did not. But he ended up working on this theory that he called the association induction hypothesis. And so he published like a book in 1964, 1965 on this, and then he published another book in like 1984 or 85 on this. And his 1980s book is like an excellent, like one of my favorites. But it comes at this time where like biology hasn't yet been like settled yet, right? So like Francis Crick, like, they're just figuring out what the structure of DNA is in the 40s, like in the 50s and 60s. It's like this dawn of biochemistry. We're just figuring out, not exactly the dawn of biochemistry, like people have been doing this for a while, but you're really like trying to like figure out life. And so these like physicists who studied like particle physicists, Schrodinger, like this, they're actually kind of like hypothesizing about life now, getting interested in this. So uh, this was fascinating to me because he was Gilbert Ling's thing is describing how the cell works and not necessarily invoking membranes and partitioning for like chemical gradients, but instead saying that like your chemical gradient changes the water structuring, and that what you actually have is like partitions of water structure. And that the cell likes to get the water structure to this place in the resting state where the water is like preferably kind of this tetrahedral pattern. Right? And you, in a sense, like are storing tons of energy there. And that you then will switch to an active state. And this is like a neuron when it's firing or a muscle when it's contracting. So a lot of the time you stay there and you're not firing. You just keep this like high energy available for if you need to like send a message. But then when you send an active potential down a neuron or you contract a muscle, you're releasing all of this energy stored in this like water lattice by collapsing it with salt ions. And so the salt ion changes this local structuring in such a way that leads to this like collapse of this. And then this leads to action and motion diffusion. And then you pay energy to reset from the active state back to the resting state. And so it's a complete like reimagining of biology where he says like the important things aren't like the membrane partitioning of environments and chemicals just bouncing around, and that it's much more active in how these different components are moving around. And so like when you have ATP, the most numerous molecules in your body by number are water, potassium, and ATP, according to Dr. Ling. And then like macromolecules, like I'll follow this up. But what are water, ATP, and potassium doing in your body? And so there's a few like fascinating insights that come from this with me reading his work that he turned me on to that I think were not taught to me before and I didn't come across, but are like really core, and why it was so captivating to come across his work was how he made me reformulate this world that I was an expert in. Like I had been doing biochemistry for like 10 years now, like at a high level. And so to come across this book written, I mean, ostensibly like thought of 50 years ago. And what's crazy is that the association and induction hypothesis is actually credited by some of the creators of the MRI as being the inspiration for looking at with NMR, which actually looks at the hydration environments, right, of your like the water in your tissues, and this gives you contrast for different tissues. So this idea that Ling was putting forward of different environments of water being central to biology, like built the groundwork for many people then to like interact with that in different ways later. So then Ling begins to frame out how these different water environments interact with each other and what their behaviors are, and how ATP and potassium greatly alter these. And so some interesting things are like the difference between potassium and sodium. Potassium is inside your cells and sodium is outside normally, right? Sodium's in the blood, but potassium's inside the cell. And I always wonder, like, could it be another way, right? And it seems unlikely because potassium is larger than sodium, so it spread its positive one charge out over a larger surface area, which makes it weaker at attracting attention of the negative charge of water on its oxygen. And sodium is the opposite because sodium is smaller. It's much better at attracting water's attention. So water would rather hang out and bond with sodium than another water molecule. But water would much rather not bond with potassium and bond with another water molecule instead. So water will leave the environment of potassium, but it stays anchored around sodium. And this is dramatic because this is, again, that shell of water. It's looking for like different ice structures around itself or something like that, or interpret as that. And this actually leads to like a different diffusion rate. And so like water in a potassium chloride solution diffuses faster than water in a sodium chloride diffusion or the sodium chloride system. And there's all sorts of like details here. So even at the basic of like a +1 ion in the cell, now Gilbert Ling gives this framework by which it has a dramatically different chemical effect and behavior. And that our cells are very much using this behavior and this difference to do work. And that, so now when we're thinking about health and disease, I'm thinking about how do I keep cycles in my cell that keep it in a healthy way to then like do the action it needs to when it does. And how do the stresses and the things that are unhealthy then take my cell away from its resting normal state or potential where it can't go through these cycles, right? And so now this is like transforming how I'm thinking of like medicine, how our cells entering and exiting like the states that they need to be in the hydration environment. And essentially, this is also what Michael Levin is looking at with bioelectricity, right? Like bioelectricity is somehow a manifestation of like potentially energy that you're storing in like the water or in the molecules in it. And so how are you distorting the local water structure that's leading to this grand control over gene expression? And some ways you might think about this is that like for a DNA or RNA binding protein with transcription factor to change the DNA that you're expressing because you need to turn into something else, you need to respond to environment. You can imagine is that for this protein to come and bind DNA, in that binding, you need to release the water between you two, right? And so you can make this place where when you're in a stressful environment that encourages these two proteins to then come together, you can just by changing the freedom of the water in the cell, bring these two proteins together, move them apart, right? And so it doesn't have to be like a signaling cascade. It can just be like dehydration, which now is so scarce that the water between them is just more favorable to leave. So they hang out together, right? And so now we can start to control cell behavior, not by like all these like drugs or transcription factors, all these little details, but just by osmotic pressure and bioelectricity. And that with these two things, and there's experiments that show that osmotic pressure really influences how stem cells differentiate, like which kind of cell they like to become at the end. So like osteoblast or bone is like much harder, right? Like much more osmotic pressure will
is
favor
the
that.
idea that osmotic pressure is just well,
Changing protein interactions?
or no, I was going to say changing the compactness of the water, essentially. Like you're getting more of this relaxed, less dense state, or you're getting more compact versions. Is it altering that?
Yeah, that's what I'm like thinking about right now. And it looks like it's like very similar to pressure. It's almost as if the chemicals that you put into water bring some amount of positive or negative pressure with them, right? And so if you think of like another crazy response of water responds wildly to pressure compared to almost every other version. This is because like as you want to expand to become a tetrahedral ice cube, you have to like fight against pressure, right? And then so if you increase the pressure, you actually like break down the desire to like build that, right? So then you can imagine as like salts as being the kinds of things that encourage that formation or like discourage formation of tetrahedral water. And this being like that main thing. So potassium's inside your cells. Potassium doesn't like to interact with water as strongly. It would rather like the water would rather leave. So water will go be tetrahedral in your cell. But then as soon as sodium is in, because water likes to interact with sodium more, you distort that lattice, right? And so you're no longer it's like tetrahedral. And so it seems like now we can understand why is it. So most of the macromolecules in your body have strong negative charges. Think DNA with the phosphate background, mRNA with the phosphate background, and proteins with like carboxylic acids. Almost everything is covered in a negative charge. And this is great because you can't stick together in the cell as easily if you're all repelling each other because you're negative. But then what's crazy is all the action that happens, a action potential in a neuron, that's sodium. That's a one charge positive ion. Right, a muscle contraction, that's calcium, like a 2 charge, like positive ion. It's these little positive ions that are like going to these effector sites on these negative things and then like displacing water, right? When an ion hangs out or like then touches if a calcium is absorbed to an active site, like that water's on some level stripped, right, and replaced. And so then again, to then do action potential or a muscle contraction, can imagine it as being like looking from this view of accounting for waters that are released or absorbed when they're bound. And so in the test tube, infinite water, very, very free to do that. But then as you start getting to like 100 mg per mil or like these really concentrated solutions of the cell, that free water, it becomes really expensive. It's really energetically costly to get that free water shelf, right? And so now the accounting might start to work out where the water isn't free as anymore. You have to take it from somewhere else. So when you're taking it, then this can like lead to different DNA binding proteins, right? Like it can lead to these different effects that we see, these different organizations, just by changing how free the water is to allow or discourage some types of interactions.
This reminds me of, I think we should take a break in just a second, by the way, just to fill up the coffee and go to the bathroom, if that's cool. But it did remind me of something I wanted to bring up, which is like several years ago, we started researching about potassium intake, right? And we discovered, and there was like a number of pretty solid studies indicating that most Americans were not getting anywhere near the amount of potassium that was necessary. And of course, sodium has been kind of a bogeyman in the room for a long time in healthcare talk and so forth. And it just makes me think about how that might play into this story in some sense as well.
Well, I think that because the statement that we uncovered was that it was less important to lower your absolute sodium intake as much as it was important to maintain a high ratio of potassium to sodium. And I think that they were looking at ancestral diets and they were like, there's a ratio of like 10 times more potassium to sodium. And that seems to be the place where you have the healthiest.
It was like a crazy amount.
It was
like,
a crazy amount.
It was crazy.
I remember reading
it.
It
was like 2,400 milligrams a day or something.
Well, because the RDA for sodium is like 2,500 milligrams.
Oh, is that what it is?
I think. And so basically, if you eat 2,500 milligrams of sodium a day, then you have to eat 25,000 milligrams of potassium a day in order to maintain the necessary ratio. But like nobody's actually eating that much potassium. And so they were like, sodium sticks around in the body for a lot longer. Like it's stickier than potassium.
By the way, like the potassium RDA is not what we're referring to either, because the Harvard article was about how the RDA was insufficient by a long shot.
So what's the RDA for sodium? I see that
it's like
you have
what you said, somewhere near what you said, 2,300.
Yeah, 2,300. And so basically, the Harvard article was like, look, at 2,300 milligrams of sodium a day, there's no way that you can get enough potassium, because that's like 23 grams of potassium a day. Like that, you'd literally just be having to add like tablespoons of it to everything that you're consuming that day. And so they were like, it probably is wise to lower your sodium intake because it's so sticky and you actually need a lot less of it than you think. And then to just supplement on top of that with potassium. And so how are you thinking that it feeds back into this? Do you have a picture in your head?
I was kind of like wondering if Michael could like adjust that. I never like totally understood the mechanism by which that would matter, but it seemed like just from the observational studies and the ancestral studies, like that we weren't getting enough potassium. And I started supplementing. We eat a lot of potassium salts in our food and so forth. And
I just stumbled on a Ling.
I was just convinced by the research that I read just observationally before, but I didn't really understand the mechanism so well.
It seems like Ling is onto this, because there was a paper that I just stumbled onto where he was like talking about, the association induction hypothesis, a theoretical foundation provided for the possible beneficial effects of low sodium, high potassium diet, and other similar regimens in the treatment of patients suffering from debilitating illnesses.
There you go.
One last thing before we leave you to the rest of the conversation is that we have our upcoming Demysticon conference that is April 7th and 8th in Austin, Texas. If you have not bought tickets yet, buy your tickets now because there are only a few remaining. We also have live stream tickets so you can watch talks on the day of instead of having to travel all the way to Austin, Texas. Tickets are for sale right up here, and we hope to see you at Demysticon 2024, hopefully the first of many.
Yeah, I kind of play around with this idea because I had always wondered why, like, at some point in my graduate career, and I don't know where this went. I think it was like kind of like, it's still up in the air in the literature, but it's like a 4x like ALS rate in like pro soccer players or like professional athletes seem to have like a high like ALS.
What is ALS, by the way?
It's a motor.
Some sort of neurodegenerative.
It's a motor disease? Like neurodegenerative like muscular disorder. Gotcha. That like, and so I think people tend to lose like extremities and it like works up the brain, and usually you like die shortly after the onset. It's that like famous Stephen Hawking had a very rare form.
It's like a demyelinating disease or something.
Yeah, it's something like that. And so I don't know like too much about it other than like usually amyloid fibers of like alpha-synuclein are associated with it, or like some proteins. So this is like my association with it. But I like know very little, and it's like reasonably rare. And usually like if it's not something else, like it's ALS at the end of the day, which means that it's like that thing where like if you can't figure out what it is, then you kind of like end up as ALS.
Like irritable bowel syndrome or something.
Yeah, right. Like if you can't figure out what else it is, it like ends up as something. Yeah. But I always wondered, like, I don't know how good that data is, but it seems like in like athletes where you sweat a ton, right? Like pro soccer players, like marathon runners, like this kind of thing, where you're like really moving through those electrolytes. You seem to be like prone for this very kind of thing where you like tend to lead towards amyloid formation. And like my thought was wondering if it is kind of like a sodium potassium imbalance, because it's so important for keeping this like necessary water environments in your cell. And so much of Gilbert Ling's stuff is like what's the mad genius of Gilbert Ling was how vast and how far he read, right? Just as Anastasia was bringing up this like paper that seems like, want to harp on this from him, is that what his like true brilliance was like reading is just, he like was all over the place. He's like pulling all these different concepts together and trying to like piece together this story that was just so big and so hard. And I think that's why his books are so hard, because it's an 800-page like biophysics paper. It's gnarly. Like, this was an endeavor when I tried to like read as much as I could, but it was fascinating with how he like pieced all these together. But yeah, just that like platform where he really tries to like build from it and see take in examples from around the world in biology that he saw and try and like frame his stuff must match that. And so at least these predictions that really heavily like seem to favor like being in the right cycle, being aware of like consuming the right amount of like nutrients, having your body go through these cycles appropriately, as opposed to like a catastrophic drug at the end to like fix your water thing, right? It's like what he seems to highlight.
When athletes are taking those electrolyte solutions, are they high in potassium also, or are they primarily sodium based and chloride?
Potassium and magnesium.
Potassium also, and magnesium.
Yeah. It's really like.
So there's just something about the cycling of it, like having to get rid of it and then take more in. Is that the theory? That becomes damaging.
Sodium is really expensive to hydrate, and so I think that sodium is just really particular. Like, it demands a lot of attention to like move through. But potassium is like, potassium is almost in a sense like greasing the wheels. I mean, it kind of like it alters the water network into a way where it's like helpful for everything to seem like move around correctly. And like when you start to get too much sodium in the cells, Gilbert Ling studied this extensively, was the exchange of like the diffusion of sodium into muscle cells and frog legs seemed to take much longer than potassium diffusion did. Right. And so what is it that's preventing it? And the thing is that he cuts open the cells and exposes the ends. So it's not like receptors. It seems to be like diffusing through the cytoplasm. So the very nature of the cytoplasm seems to like select against it. But once you do get sodium in there, it kind of destroys the whole, like you destroyed that local environment. Like the ATP is purged, like stuff's changing. So I don't really have a good like mechanism, but like there's a lot of interesting things here that are unresolved that really point to it by his work. Of this being really interesting and important.
I mean, I think there's a lot of really interesting stuff there, and like just like we're at the edge of any science where we're really pushing into the unknown, we shouldn't necessarily expect a bunch of answers right now. We just have a bunch of questions. But the thing that really intrigues me about this, and it might make a hinge into talking more about consciousness and the human story across the ages and so forth, and your introduction to a lot of this thinking through the UAP business or the UFOs, is that we're looking at an interesting paradigm where, actually I majored in neuroscience as an undergraduate, so I was also like looking for this mechanistic explanation. Like I thought of it as a harder science to study the human behavior, right? That was like my naive vision of what that would be as a discipline. And you start thinking about, okay, like consciousness is a production of all of these interlinked neurons, sort of like a computer with all these electrical signals going on. But what we're staring down the barrel of here is biology being there's this whole dark matter, which is water, that's operating in the background. It's also electrically complex. It also seems like it's capable of causing behavioral changes, right? Structural changes, like action, let's say, of the organism. And so it's like, my God, is there like a dark matter of consciousness involved in this water structure as well, right? I mean, look, I'm not saying there's any evidence for that. I'm just saying that it has all of the cabling, let's say, for that to be possible, right? And so you start wondering if, going with Levin's idea too of consciousness being distributed, like not just being a thing that's made by brains, but being throughout the biology. And then like you almost border up at this point when you start thinking about water as being as having a potential role in experience and action. So consciousness, as far as I can define it, then you're basically brushing up against this almost Gaian articulation to the cosmos in general, where like water is now actually a somehow participatory in the conscious experience that we're all a part of, and that we're looking for the source of. Like the reason why we're studying neuroscience or biochemistry, right, is we're trying to understand how the hell this happens. How is it that we're sitting here, we're a bunch of atoms just like having a freaking conversation across the internet? Like it's nuts, right? That's,
Right.
I mean, I think that's the source of a lot of like early scientists trying to look at the world and be like, this is nuts. Like, how's this happening, right?
Right, like where's this hiding or not? Yeah, I think that would to me was like particularly fascinating because as you kind of like come up against is like water has almost this like mystical like piece or like this dark matter, right? Like we have this thing, we have like this measurable object, but we're not measuring enough, and like does water fill that? And that was totally my like interest and kind of what I like shared earlier about this interest in the Orca-War, this theory of consciousness proposed by this like Nobel Prize winner in physics, not for this, but Roger Penrose. And he came up with this idea of he needed like a way to like collapse the wave function to like manifest reality. And he came up with this kind of like quantum computer idea. And this American anesthesiologist, Stuart Hameroff, said, hey, that sounds a lot like you're describing a microtubule. So they started to work together where it seemed like a microtubule is a cylindrical like protein filament where it's made of all these like small globular proteins that hang out together and they build this huge tube. And this is part of the cytoskeleton, the skeletal structure of your cell. And it's like really complicated. There's like a lot of things that happen on it. And in fact, like a microtubule binding protein tau is a famous Alzheimer's protein. So I've been like coming across this for a long time, but they had Roger Penrose and Stuart Hameroff proposed that how aromatic residues are arranged in the like helical structure of this protein filament can act as kind of like qubits or in some way be doing like quantum computations. And again, I can't like interact with much of that, but the defense of this theory, there's a lot of protein biochemistry where microtubules absorb a light almost as efficiently as like photosynthetic harvesting proteins. And so they're able to like absorb and interact with like wavelengths in odd ways that I had like no handle on. And what this became was this like moment in my mind of seeing kind of like how these structures with the water around interact with light in a very particular way. And so it was almost as if like I was trying to like find out this like transform of how information from light got turned into information like movement and sound. And this is how I kind of like start to appreciate these kind of like more esoteric or like new age things like light frequency vibration. Like what does that like actually manifest as? And I kind of came up with my own like heuristic because I don't have a strong physics background trying to like what do all these things mean. It's like everything is kind of like an antenna that can accept, and like every antenna is a transmitter as well. So you can like send and receive, right? There's no real difference. And so every atom, every particle has like different resonant frequencies that you will like interact with, accept or emit. And so when you accept light, then like if you know might be an electron transition where electron like jumps to a different orbital and then like jumps down, you emit that light, and there's a difference in like the absorbed and emitted wavelength. And that is actually released as heat and motion, right? So when you're absorbing light, there's a motion associated with that. And when you release it, there's an emotion associated in a molecule to release it. And so whenever light is absorbed with something, there's an associated physical sound, right? Motion and heat is sound, right? It's how like motion is moving through matter. And so every light wave, if you happen to have like the right frequency, then you like produce a sound or a motion in the physical world from absorbing and emitting that. And so frequencies that transform from the domain of light into the lane, like area of motion. And so it then like strikes me as this like microtubule structure is this wonderful like transform, right? Of having all these pieces where you can turn information from light into physical movement of like sound and motion. And so like, how do I like move my arm, right? I have to like build this crazy Rube Goldberg machine that I can like with a small motion in my brain somehow trigger a vast motion out here. And so it became kind of curious of like, what if like light was one of these like forms of transmitting this information, because I was exposed to this like wild biochemistry that I never really appreciated except through these physicists trying to figure out how it's being involved like consciousness. And this was beautiful for me because I remembered in that moment coming across these like seminars that like, oh, I was super interested in consciousness. And I think that water becomes this really important like transform. And I earlier talked about like going this two-state model, like going in between these like two different states and storing energy. And I don't know how this all like works out yet, but that's like the true mystery and the fun part that is for me. But earlier, like Shiloh was mentioning how, is there like room for mysticism? That was definitely like my interest. Like, where is like the secrets lying in this water shelf, this like transforming information of light interaction, transporting it, and like turning it into like protein motion. And so the more I like studied it, the more I studied water, it became like less mysterious and more grounded. I found this like kind of fascinating because it's almost like I'm into it because I'm so interested by like the potential is there this like deep buried mystery here? And the more I interact with it, almost the more mechanical it becomes. And there's this like, part of you kind of like dies. It's not like, I don't know, not something like mystical or interesting in that way, but it also becomes like really satisfying in seeing this. And this actually ties back into the Pierre microwave background question, I think earlier, because the fascinating thing about hydrogen bond is that when you're doing like van der Waals interactions, you're just hanging out on the surface of another particle, but you can't like go into because the electron shouts are repelling. So there's no like overlap or sharing of electrons. And that's what chemical bonds are, really, is like chemical bonds are a sharing of electrons between two atoms, and changing chemical bonds is like rearranging how you share this stuff, right? So then what's special about the
And it seems
hydrogen
like you
bond?
need that shared, it seems like you need that sharing for a black body profile to happen because you need
Yes,
all those overtones, right? You need different lengths
right.
of, you need this whole array of possible lengths of your antenna to get all those frequencies, basically. You can't just
And
have a
now
single antenna
this is
like a
what's
gas.
exactly crazy about water is that water, if you look at it in the gas phase spectrum, has nice like peaks
Right.
for all these like intense, all these absorbances. But as soon as you get into liquid, these become really broad. It's almost like it becomes that black body absorbing so many. So my theory for where the trick is on this is that the hydrogen bond is actually like a 15% overlap. Like a hydrogen bond is 15% closer than van der Waals interactions would be. And so this means that a hydrogen bond is special because it's a pseudo-covalent bond. And there's a very technical way of saying that the electrons have orbitals. They're not just like a ball around you. They have paths. And since these paths are like directional, that a hydrogen bond that's straight is far, far stronger than one that's bent because now the orbitals like aren't aligned appropriately, right?
Mm-hmm.
And so this is kind of like, we might
They're
call it
just less
like
enmeshed, essentially.
they're less enmeshed. And so now you have like a way with these two-state waters where you have tetrahedral water, you have larger antennas, right? You have an array of shared continuously in many directions, shared electron clouds, right? And so then as you start to like destroy that tetrahedral structure, you will change necessarily seemingly the absorbance, the frequencies that you absorb and how you do, and the different salts that you do will change this in different ways, right? And so what we're really doing is we're playing with how that structure that we could describe as like an antenna of absorbing or emitting very particular like wavelengths of light, but kind of as a black body because it's so like or metallic bonds, right? Where you're sharing electrons so far. And this seems to be like one of the true beauties of water, of like having some of these. And so this is something that's becoming like really appreciated in the literature in the past few years as we can get like really fast laser pulses and really, really interesting ways of measuring like water's behavior. But I think that is like water is this wild, like semi-continuous network of overlapping orbitals that allow for crazy communication, also crazy absorption of like way longer wavelengths than the one molecule, right? Like uh,
He makes
microwaves,
this
all
really.
this,
There's a
right?
point you might check out too that Pierce made a few times, which is about these nuclear shock, these nuclear tests, and you can see that like when
Yeah.
the. Did you see this in that he?
No, I
I mean,
haven't
he said
actually.
yes,
I'm
but
really curious about this.
well, he just points out that during the bomb tests, like you're probably watching like that's crazy, so like you see these little ships like bouncing around and so forth. But if you look carefully, you actually see that there's this ring of black that spreads out from the bomb explosion in the water that comes all the way to the shore, just a really black, completely opaque ring. And his point is like the compression of the water is altering the lattice structure, much like what you're saying can happen in all of these different parts of the cell as well. Like you're changing that, and it changes the optical properties as well, which means that it's changing like the conductive properties and everything electromagnetic that we would expect as well.
Yeah, and so like when we're talking about like easy water from Dr. Gerald Pollack's ideas and things like this, right? Is like large like forming like a large network, and that's what a lot of times what it seems to like be measuring or describing is this like region or boundary. And so like if you're, and essentially this is like nicely organized tetrahedral water. There's ways to like describe this that he does as well. And then so he uses naphion, which is like a negatively charged polymer, like gel to like build these surfaces. Well, microtubules, they have tails that are all negatively charged, right? It builds again this like shell of negative charge polymers around itself to seem like structure water. And then what happens like when you have a neuronal impulse and you have a neuron with a bundle of microtubules in the center and like the membrane on the outside, you let sodium in and that tends to collapse, right? It's like a pressure sucks the water up. And so now you can re-emission as opposed to like a voltage change is like a pressure wave through the cytoplasm of a nerve being the thing that's transmitting the information, right? In addition to this. And so there's like that physicalness I think Pierre Robitaille is talking about. I think that your cell seems to build this water network to then collapse as like carrying this like wave. And the cool thing about that is that when it hits you, it's like the water environment is different. It's going to tug on the proteins differently too, right? Like there's all this like actionable, like interesting physics that's going to happen when you do encounter one of these waves. So I think that something like that would exist in the body, and it's fascinating that you share because again, there's like too much of this to connect all of myself. And that's
well, that like border between physics, which is essentially math, right, in the modern world, the border between that fuzzy math abstraction and like where the rubber meets the road, where the cell's actually doing something, that's the frontier as far as I can tell. Like that's where so much work is left to be done. That's kind of what drew us into the fold. But maybe, maybe you can switch because I want to see how in the heck this leads to UFOs.
Right. So this whole thing came up because I was like curious about the UFO story, like with me for a long time, kind of growing up interested in that. And so in 2017, there's the New York Times article that kind of had like government whistleblowers, UFO programs. And I remember being in grad school and reading that and be like, this seems big. I'm sure that like the scientists will figure this out in a few years. And then when the pandemic happened, and I like thought that I remembered this and we're like, wait, has anything happened with that? And so I started to like look it up, and I came across the Theories of Everything YouTube channel with like Michael Levin, all these people that exposed me, Stuart Hameroff, right? All these
Kurt's been doing a lot of work in this space. Like he's pretty much like in that cartwheel rut.
And the beauty of like what you guys do and what Kurt does is that as like a practicing scientist, usually what I do for new information is like read a journal that I've like or look for articles that I'm like familiar with. But the cool thing was like I'd be exposed from all this other like information through my interest in UFO and like listening to these government whistleblowers. I was exposed to all these scientists talking about the same thing. And the beauty of that was at first it's like discouraging. It's like, should I study like UFO physics now? But then it's like, I have things to contribute with my own interest in like the biophysics stuff, with the water story. But then the outcome was my interest in UFOs kind of like led me to this like the interesting like consciousness stuff, and also like Gilbert Ling's work, and Michael Levin's work, which became like immensely influential in what I look to do every day as a scientist. And what was really wild to me is this like really pulled off the veil of like Ling's work is like ancient. It's like 50 years old. And I found so much wisdom in reading and engaging with it. Even if he's wrong, just in how he thought about and framed with it really challenged me to think of new ideas in like biochemistry. And I found it so creative, but it seemed like his work is like largely ignored for like many reasons. But I think that we lost the wisdom that he put out there. So I, within my own field, uncovered this like mystery school of biochemistry, right? As opposed to teaching like membranes and things bouncing around until like reactions happen, this totally reimagined way to get to the same place, right? And that get to the same ending, but really highlighting different things that is like holding dear and elevating as to like build this theory. And so, of course, like reality's somewhere in the middle, like everyone's right about something. But I think that I was then, as someone like in my field, like my peers are not like paying attention to this, and that's because like they have like important things to do in their world. They don't necessarily like need this yet, but I needed this. And I like didn't know where to find it. So it was like interesting to me that like entire fields and schools of knowledge could like not be passed down, right? And so now I'm getting to the UFO world,
which seems to be a pattern too, right? It seems like the further you dig into the history of science, which is a big part of what we do on this show, the more you find that people have had really revolutionary ideas that just get lost, sometimes for thousands of years. Like the atom basically got lost for 2,000 years, you know?
Right.
It's like, how in the world is that possible in today's
Right.
modern information landscape.
I agree. And so part of my like disbelief of a lot of UFO stuff as like a skeptical scientist interested in technology to, build something interesting, understand something more about the world. Like what's the most out there creative place to get ideas? Probably anti-gravity UFO, right? Like let's do it. And so looking at that stuff, I was just so fascinated, but it became really, but then simultaneously going through the water story, I think a lot I had unveiled for myself that like important information can be lost, and that even though I might expect for people to like have this behavior, they don't always have that behavior. And so much of the UFO stuff is looking at a whistleblower. There's no hard evidence. There's nothing I can dissect and like measure myself. It's all taking a story. And so much of these stories, you're like, I wouldn't do that in that situation, or like, or wouldn't someone do this? And so we find these ways to disbelieve someone because we tend to think if like I were there, I would do it differently. And
Give me an example of that.
I tend to think like how my own experience with this was as I started to take seriously how like the level of government officials that were talking about this, and like we seeming to like with great sensors measure things doing like physically absurd things, right? And so like take that and
Like the
that's,
TikToks or the TikToks?
the Tic Tacs,
Yeah.
right? And so then like there's the news releases, there's the videos we've seen, but then there's also like the people who release that stuff like years before, right? Now so I'm like, oh, so people have known about this before the New York Times. That was leaked way before. Who is doing that? Well, it's this guy. Why does he have access? What else do they say? And so the Tic Tac stuff, I was like, that's interesting. But then the person also released like a phone video of radar contact from like that some sailor was like filming at the same time. So now we have like two sensor suites, right? We have like radar and we have forward looking like infrared cameras. And that's what we're like really measuring, like missing is like, how can we measure from something from 2 points? Because when you're just listening to a human story, it's just one vantage point, right? And so in science, we're always trying to like get multi dimensions at the same time. And this is how we're like narrowing down water's behavior, like anything we're looking at. And so I was from this, I don't have access to the originals, but I now tend to believe that people are kind of like telling their truth. I don't necessarily think that these people are like, why would this person be filming like something absurd on the boat he's on because he's witnessing something absurd that he's on the boat he's on? And I just like believe that like our sensor suites are measuring things, but I can never verify or check that data. And so I can't like touch that really. But then it opens up this thing. Well, for years, people have been saying things about alien abductions, right? And so I started to like look into like alien abduction and cattle mutilation. And this is like interesting because I feel like cattle mutilation, there's a lot more actual like good data of like FBI agents in the 50s like having to go like interact with people complaining about this, or this like showing up you can like have the carcass of the animal and you can like measure certain things. But again, so it's all like personal stories. So when you start to hear people describe like UFO experiences on like YouTube channels or different things, and of course it's but they seem very similar, and I kind of like judge against what I like knew as a scientist. And so a lot of them have like these similar forms where someone will describe, and so like I'll describe like what they say, and then like as a scientist my kind of reaction to it is like I say like awoken by a light or something, and then like told by beings walk with them like into their craft where they do like experiments on me. And so it's like when you're like, what kinds, so like the possibility of all this, like what kinds of experiments are they doing on you? And they would respond like genetic experiments. And so, like to me, this is hyper plausible, right? Like abducting a small mammal and shooting DNA into it is like a very common experience in the world that I inhabit, right? And so like
it seems like definitely a very human thing to do,
right? And so this seems so like familiar. Like I have no issues with that part of the story, right? And
in so much as those things are like us, right?
In so much as it's also something that scientists do. Like scientists are doing, like you have a colony of mice and it's dark and all of a sudden lights come on and a hand reaches in and takes a mouse and like does a bunch of stuff to it and then puts it away.
Right.
Like that's a reality.
This isn't the tall tale.
Right? Yeah, to some degree. Like the tall tale is the fact that there's like somebody larger than us who's doing it. But if you're able to see it on the level of us to mice, it's not that far of a jump to be like, well,
Right.
This is a thing that
That's
advanced
But then that
species
opens up the
do.
door to like projection, right? It's like, well, this is what we would do if we were studying another life form.
that's
So
exactly like a prison, but that's where I was at this time, was me, the scientist, trying to interact with like the available data to find out what's like good there. What to me was like good and useful. And so part of it is like that part like makes tons of sense. Fine. And so I had all these things I call them like jumping off points, right? Like when would I like turn off the show? Like when would I stop listening to the story, right? And so it used to be just like, well, so what else did it use? Like, oh, like probes or different like devices, like that makes sense. Like how did they like wake you up? It's like there's a bright light outside. Like I've seen like I'm not too worried about that. Like how did you get downstairs? And they'll be like, oh, I walked, but they seem to like float. And they're like, that's like weirder.
Well, there's
It's
a
like,
lot of stuff where people feel like they're passing through walls. I've been reading John Mack's
yeah.
book.
Oh,
And
that's great.
in the abduction experiences, a lot of the times it's people pass through surfaces, which is like
That's
a
right.
little bit weirder. And they're like, I felt a faint vibration as I passed through the wall.
That's
And I'm
right.
like,
And that's what kind of talking about these like jumping off points was asking too, is like then will like people will talk about like going out in like a snowy day and like they can see their footprints, but none of the others, or some, or these things where like it seems physically impossible. And another thing that comes up a lot is like telepathic communication, right? Of where you get these and this used to be like hard for me, and this was hard because it just seems so hard like to imagine like an alien being totally different evolutionary background or makeup coming here and being able to communicate in the same way is just so absurd to me, because it seems like so hard within, right? Like, does this like evidence of like a universal language? There's so many things it could imply, but there's also the nuts and bolts, like, is this happening?
and like if
it
we
was
have the
all-
hardware for that, why don't we use it or something like that?
Right. Like it seems like we'd have like known about this before. It seems like we'd have known about this before, right? Like again, this whole thing is this like refrain that kept coming up against it being like, I feel like we'd have known about this. So like going back to the tapes, like what do we know about like telepathy and this? And so this became the most convincing part to me of like the whole UFO narrative, was that there's no good like data at the end of that tunnel, but there's very convincing stories by very convincing people. And some of these things like really like fascinating. I would cause me like take these people and these things seriously, but I can't like say if there's like truth there or not. But when
well,
we get
it's
into
just,
like
it's a little bit, for me, the hardest piece of it is that I'm like, if this is a species that is actually doing something to us humans, did they just start doing it recently? Because
yeah,
these specific narratives seem to belong firmly to the 20th century.
right. But then Jacques Vallée's whole contribution to this field is then trying to continue how these stories mirror fairy tales that we inherit, and then extending these stories as like a modern retelling of things we've had for a long time.
Sure, but then that makes me feel like it's more like schizophrenia than it is a real event, because that's also what
Yes.
schizophrenia does, which is, it didn't used to be that you had fillings in your teeth that you could hear the CIA in. It
Right.
was angels and gods, and so it's the same. It seems to
You
me
can
to
also
fill the same
you can also
category.
hallucinate without being schizophrenic.
Well, no, I'm not saying that these people are schizophrenic. I'm saying that like the quality of the hallucinations follows the same sort of cultural influence that schizophrenia seems to follow, where
Mhm.
your background defines, or your culture defines, the sorts of hallucinations that you have as a schizophrenic. Does that
Yeah.
make sense?
I think it defines like people's meditation experiences or psychedelic hallucinations, like all sorts, right?
Yeah, and so I'm like, doesn't that imply that it is being formed internally rather than externally? Because if it was external,
I think yes, but it doesn't discount it from being a genuine phenomenon. Like it's like, so what is this?
well, so but as long as yes, but the phenomenon then becomes an internal phenomenon rather than an external phenomenon.
Not
Right.
necessarily.
And as a scientist, like I feel like either way there's like a there, right? Like either way I like would like to know the answer. And so I feel like kind of big takeaway for me is like people are having like extraordinary experiences, and if it's not like in physical reality, they're still like wildly changed from it. And this was John Mack's big claim to fame, right? Of like how he was like prohibited from his practice for a while, but at the end of the days these were people suffering from severe like trauma and distress, and he was the only one talking to them. And that was the only like true message I think of this whole thing, was how like people got better by talking about it and not by not talking about it. And, like that's the only like real takeaway from like science for me at this point, right? Like is that it was so much more powerful and productive to like really listen to these people as opposed to like jump off at my jumping off point, like turn your channel when it got like weird. And because like it became less to me about trying to like figure out the nuts and bolts, but to try and understand like the consequences for these people's lives are severe. Like they quit what they're doing, like they quit their government job and become a whistleblower. They like quit this and like to do that. They like I was sort of someone who was like a lawyer in their 30s and then like had a vision from an angel. Like someone wants to like really quit their job and like have to deal with that or like unmask.
I mean, I guess I
reality,
could be skeptical. Like if I put on like my tinfoil hat, I could be like, well, they're
they
still
really
working
didn't
for
want
the
to be
government.
there.
Or they're still like they're just doing their job, you know, their job is to
Yeah.
pretend to have this experience. Or you know, I think being a lawyer is probably super stressful and a lot of law is really mundane. It's not like, you know, paper chase stuff for the most part. It's just
It's true.
like filing
And so either
paperwork.
way, like
and
there's a there, right? If we were like causing people to have like mental breaks that have like angelic revelations because of our culture, like we should probably know about and like take that really seriously as opposed to discount this person, right? Like no matter how you cut it, this is really worth their time and attention.
yeah.
And so I was originally interested for the nuts and bolts to get the technology, get that good data from these people who had like those nuggets to like make scientific breakthroughs. But then I kind of realized like
There's nothing. That's the thing about saying that there's a there is it becomes really tricky because you start to listen to these accounts and there isn't actually if we're saying that a there is a physical, tangible experiment that you can do or data that you can collect, there stops being a there because the there is the story. The story is deeply internal. And when you start to actually try to collect some sample that you can test and be like, okay, so this is an implant this is something that came out of your nose a week after you had this experience. They look at the object and it's like, well, there's nothing unusual or strange about it. It just seems like a piece of metal. And it's like,
yeah, but it was found in your body.
it was
like,
found in your body, but it's like people are also, you know,
and this
You
know
can
the,
talk yourself out of all these things.
So
so-
more
this is
like there's
exactly this is why like there's no evidence in the UFO world I can convince you of anything, and that's why I just like got to like enjoy hearing people's stories and what they're trying to share with me, and like leave it. I like kind of cut my ties to trying to find like a there because I think that the there for me became like, you know, at the end of the day I'm like dealing with people, not atoms, right? Like and so I like shift my focus.
and there's
but at
only
the same-
so much that you can say. It's like, okay, well, you had this experience, and that's important and that's meaningful, and that's a there. But it doesn't rise to the level of like true scientific inquiry, which is like, okay, we have an object at the very least that we're
right.
focused
But
on.
if like a measurable result is like how we move atoms around the world, these people have such fundamental changes in their lives of how they like move and behave, right? Their atoms are doing very different things than before, right? They're like maybe not well and they like really need help and it's like a cry for help. Like their atoms, like we need to take care of that. Like even, or maybe they're like the opposite, like it's in the best of intention. Like there's dramatic consequences into our physical world with how atoms are arranged by personal inner experiences, right? And this is like the there to me of like it's always worth like me paying some attention or like trying to understand what's going on. But again, like there's no data I can like convince everyone of like UFOs, but I will say like the American government, like there's a YouTube channel called Red Panda Koala, where this person like really catalogs a lot of like history and in-depth like history with the UFO phenomenon in the American government. And so you can kind of like you listen to these stories and be like, okay, there's probably something odd with the American government interacting with this. And so the only like receipts become like odd interactions, and this kind of feels like some distrust. And there's like more of a paper trail, so it's more physical than the UFO technology stuff in some way. There's like a history, but then it is the idea of this like telepathy that stuck with me, because this is something that's like much more measurable scientifically, right? And this is something like Dean Radin is like famous for, of a professor like Princeton Engineering Anomalies Research Institute, or like there's various other research institutions that seem like relatively serious and like relatively diligent at are trying to interrogate these things. And so the first thing is like, can you just like prove that it exists? Like any like telepathy or that? And the answer is like, I can't. It's like you have to prove it to yourself in the sense of like, I don't know what's the information that's convincing to you. But what I had for me was stories of like past life remembrances from children that are born in and can say that they have like memories of being a different person, or So there's like a famous one that Leslie Kean mentions and is on her like Netflix series, I think like Life After Death or Surviving Death, about a young boy who recalled that he had died in World War II in a fighter plane, and they found like his unit, they found the specific person he was, like in the crash. So there's all these things where these
He like
children,
remembered the name of the person that he died with, I think is what it
yeah,
was. And so it allowed them to triangulate
right. And so
what
he
it
actually got to
was.
like, yeah, they tracked down other people that were in this person he remembers flight squadron that were old at this point, and you got to like interact with them and remember all these details, like plane types, like all these like bits that it's not impossible for like a 3, 4, or 5-year-old to come across this information. It's just like really,
It
it's
stretches the
unlikely.
imagination. Like you'd have to have a
It does.
conspiracy where like the parents are feeding him this and then
Right.
he's like
Your
playing
material
into
like
it.
explanation becomes as like when it's just invent some aliens or something. But it's possible. It's possible, right?
Yeah.
So it's reasonable, right? That we can this data isn't perfect, right? It's not just like a mechanism. But to me, it's so convincing. It's so like I have to really work to make it like
Well, it just seems like
meaningless.
one of the places in science where it's obvious that something is happening, but there's very little understanding of what could be causing it. Like, I think that
Yeah.
for me, near-death experiences and the phenomena of personality changes that occur after organ transplants, those are probably the places where it's most robust, where I'm like, I don't know about UFOs. I'm really, because there's no there, the only thing that I can reach to is, okay, people report things and they seem to have durable changes to who they are in the aftermath. And it's like, okay, so
Yeah.
something has happened to that person. What
Right.
the actual
And something
phenomenological
congruent to
and
that seems to be
material
happening for a really long time.
and something congruent to that seems to be happening for a really long time. Like you change the
Yeah.
mythology, the stories are a little bit different, but like, okay, something divine reaches down and touches people and they experience it and it changes their lives. Okay, there's a there. But like you said, hard to study. Near death experiences and like organ transplants changing personalities, that seems to be something that's like a little bit more physicalist, where
Yeah.
you can look at it and you can be like, okay, well there's a procedure that we have done in a laboratory and there's something physical that we have moved from one body to another. What does that tell us about the way that these systems are connected, and how does it blow apart our narrow-minded perspective that it's like, no, the seat of consciousness is exclusively the brain and it is exclusively neurotransmitters in the brain, and that is the beginning and the end of that story, and we have closed the book and no more do we need to look at it. And so it's
Right.
exciting because as somebody who's going into the sciences, like you need something that people aren't looking at, because what are you gonna do? Like figure out another signaling pathway that leads to double strand breaks in cancer and like faulty repair mechanisms? Like there's people that are doing that, and there
Right.
seems to be an infinite pipeline of them.
Right. And so I like very interested in this like green space as you described, right? Like what's cool about like this data on like near death experiences kind of is that like like we'll call it like the frontline, like frontline researchers on the UFO world is like all over the place. But the frontline researchers and near death experiences, basically the like doctors and nurses, like medically STEM trained professionals who are like witnessing this and like reporting the data and like noticing like odd things. And also oddly enough, is like not PhDs, right? PhDs are not on the front lines, like noticing this stuff. So like we're always gonna be more skeptical, right? We're not there, like being confronted with what this looks like. Uh and
That's
so
a
Unless it
really
happens to you.
yeah.
unless this happens to you, right? And that's always the most important life-changing event. Or it's just it becomes a lot more believable then. Yeah.
I think that that observation that the PhDs aren't there is really apt, because even Mac, he was there in the sense of being a psychiatrist who was open to even talking to people about this. And so that gets you one step closer to being there, so to speak.
Yeah.
And he was open to all of these methods, right? He was doing a lot of hypnotic regression, and you can criticize hypnotic regression and say that it's something that was being produced in the session, but it also, you can plausibly say that it's an honest recapturing of something that has been paved over in the mind. And so
That's right.
if you're open to it, you can find all kinds of things, and that seems much more exciting than just abandoning
My problem
all
is
hope.
like the priming aspect. Like I've had like some similar experiences, I guess, to these near-death experiences. And even as I reflect on my own visions like that I had during these experiences, I wonder how much of them were just shaped by what I was expecting from what I had heard about in all of these since I was a little kid, and fairy tales all the way up to my scientific study of near-death experiences, right? How much am I just living out what I was programmed by my culture to
Right.
experience?
Or how much is the experience necessarily patterned onto a pre-existing cultural skeleton?
Yeah, that's kind
Where
of
it's
what I'm
like something
Yeah.
happens to you, and the only way that you can process an event is by telling a story. There's literally
Right.
no other way to understand something. Like you have to
And
have like verb, object,
yeah,
subject somehow related
right.
to one another.
Yeah, and you can only like see what you have stories for in some way, right? Like, and again, like, yeah, if you have a culture with different terms for colors, they'll see colors differently, right? Like this matters so much. So when you're like approaching anything, it's only like ever a reflection of you, of what you bring to it, right? On like some level, even scientifically, right?
Yeah, and
Like,
like fundamentally, there's if you're having a near death experience, systems are shutting down. And so
yes.
the
And so
things
or coming back on.
there's like interesting physical mechanism of like I've heard like all sorts of things where like balance comes from like the inner ear and like crystals and like muscles playing with it. So if you like relax those or something during a near death experience, you could have this idea where your center of balance is like outside your head, right? And so like a physical mechanism for an out of body experience, right? Like all of these things, again, like as you look into them, you can find these physical and again, like when I looked into the water, the mysticism died. Like it became this like interesting, like fascinating mechanical thing where there is a lot of room for like cool stuff, but like there's forces acting on each other. And so it gives like a lot of like credibility that it's like coming back with information that's unexpected from these. And so one example of this in like Leslie King's work is this woman who was like having a near death experience in a hospital, and she could like see and float above her body, and she saw like a shoe on a rooftop in that experience. And so like they went and she mentioned it and they went and checked and found like a shoe there on the top of the hospital, right? And so the things that stay with me is like I can wash away all these like physical explanations and mechanisms and like and what that, but it's this like remaining thing that sticks with me is like this ability to interact with data and retrieve data that seems like you should have no access to, right? Whether you're a child coming in with like information about past life or assuming like absurd amount of information about World War II airplanes or like obscure villages in India, like all of this has been kind of like recorded, or this experience in the hospital, like people come back with information that I don't see how they physically read. Like we our physical theories of medical and science like should have to explain this if they're going to be a theory of everything or like appropriately complete. Like we should have some way to like address this, right? And so then having to like drop down what I like consider as like information, like really explore this and like how are we getting this access to this information. And so then this really is what opens the door to me to like the more esoteric stuff, right? Is like once I'm like appreciating that people are coming back with like really actionable, testable information from these absurd experiences, I don't need like all the, you can, again, it can all be like a physical fluke, but that's still so useful. It's so interesting that you have this like information. And so, you know, it turns out that there's been like a long history of humans like interacting with like telepathic abilities or trying to like uh, carry this along. And a lot of time this isn't like, uh, you know, talked about in esoteric groups, but Dean Radin is like a contemporary scientist who spent a lot of time on this. And he, I think that there's a few interesting things from like his work to take away. Again, like little tidbits like random number generators around the world, like on 9/11, random number generators like seem to like deviate from what was standard for like the numbers they were generating. Uh, Odd things of where like when you try and do tests of telepathy between people, it becomes like this was fascinating to me of how hard it is to test this. So what Dean Radin describes is this phenomenon where like with the like Stanford Research Institute declassified CIA remote viewing files, there's this big this kind of like set of release where like, kind of like you send a target to some place and then there's going to be someone that tries to remote view that person, and then this person's remote viewing is going to try and describe where they're at. And so if the description matches like where the target was at, then it's a pass, and if it doesn't match, then it's a fail. But the thing is, there's so much room for interpretation there. It's so many degrees of freedom. It's so open. So the results were tended to be that people thought remote viewing was possible. And then there's like some counter arguments that are published against this, refuting that like from the notes given, the person like could kind of have like guessed before, or there's some other arguments against it. But when you have a lot of degrees of freedom, people tend to describe experiences where they believe they're remote viewing or getting like information. But then if you try and like refine it, well, like I just want to, that's like too broad. There's too much like experimental problem to like really get the info. So we're just gonna like have you in a room and like try and communicate like a color to someone else or something like that, right? Like just a really simple, like, what is this color thinking in this person's mind? What is this? And so it describes this phenomenon where it seems to like be good for a few tests, but then your data gets muddy as you get to like 1,000 or 100, right? And so the more that you try and like isolate your variables and hammer it down, the harder it gets to prove telepathy. And there's a few things that he poses for this. Is first of all, like this second, third, fourth, hundredth time, it's not like interesting. There's no like people are bored of that anyway, so that won't be on their best behavior or best game. And then the other thing is like as you try and like isolate the noise by allowing like disallowing like degrees of freedom, you seem to like lose the effect, right? It seems to kind of shrivel up. So if you just like narrow down, it's like, are you gonna hit A or B or something like that? I don't know. It just seems harder. And so there's some like responses to this that can take away is that first of all, like upon finer measuring, the effect size disappears. That might be it. Or it might be that like, oh, you need an emotional interaction for this information to transfer, right? What's your emotional interaction to like A or B versus like a waterfall, right? So maybe it's like the methodology is flawed to capture what we're trying to capture, right? And what I'm trying to illustrate here is that you can rigorously test this. Scientists who take this seriously rigorously test it. The results are kind of fascinating. We can learn a lot about that, and there's room to grow there too, right? Like there's more for us to do. And I think that was really like eye-opening about Dean Ray and these other people like taking this seriously and trying to like it's a fascinating set of stories for science where we're trying to like redo each other's data in this field, but every attempt to like nail down the variables that you're worried about, you seem to have these consequences where you seem to like kind of destroy the effect you're trying to measure. And this is something to be like skeptical of, right? But it's also something that is its own way data. Like, what are the kinds of things that resonate? And so, one of the most convincing and interesting experiments that they did to me was a computer screen where it's going to like randomly flash 100 images, and like 2 of them might be like very pornographic or grotesquely like yeah, not great pictures to look at, and
Shocking.
but most of them will be neutral. And so the idea is you have like a really strong reaction to something that's like highly arousing, like violent or pornographic, and nothing to else. So like you're going to see this image, and then you're going to like register in your eye, then you're going to like register in your mind, and then you're going to like sweat or do nothing, right? And they're basically measuring your body's like responses. And so the question is like, when one of those striking images comes up, how, like, at what point do you sweat, or like, does your body have a physiological response? And the answer seems to be like a little bit before the image is shown, right? It just seems absurd. It's like hard to like, how do you, I don't know, but computers got like really fast measuring. It seems like.
But is that on the first trial, or is that on the second and third trial? Do you know what I mean? Because if you put somebody through that experience naively, this is exactly what you're talking about. Like, they don't know what they're going to see yet. But if you do it a second and third time, now they know that as the experiment is running, something is coming. And so the longer that the experiment progresses, the more that they start to get prepared for the thing. It's like kind of bracing yourself.
Yeah. And I can't agree more. Someone has a name for this, right? As soon as you set like a scoring function, or you're like testing someone on something, you're no longer testing for that thing, right? You're testing for the test.
Yeah, exactly. And so this is why it's so difficult to track down, is that we know that.
I mean, the crazy thing is you've had one of these experiences.
Yeah, so we had a patron who was like, you need to do this thing called the Silva method. And the Silva method is this, it basically is a remote viewing technique that was worked out by a guy in, I want to say the 70s and 80s. And so now there's an organization that runs courses to teach people how to do this. And I was like, okay, fine. Like, I don't really buy this stuff, but I'll do it. And so the entire course is just meditation and visualization experiments where you're just going down into the state, and there's a teacher who's guiding you through seeing all of this stuff. And the course coalesces on what amounts to a remote viewing exercise. And so the Silva method, people collect all of these medical reports from people because the idea is that you can then apply healing thoughts to people who are sick, and using the Silva method, you can heal people from a distance. And so the idea is to treat these sorts of healers and for you to be able to heal yourself. And so at the end of the course, after 3 days of this, you get paired up with people. Actually, I think after 6 days. And so each of you has a list of physical characteristics, age, symptoms, and location. And you tell the person the gender, the age, and the location, and they have to tell you, when they go into this kind of trance state, the physical ailments that they see in the person. And it was really weird because you hear the age in the description and you just immediately get a mental picture. And then there's this thing that you're supposed to do with your hands where it's like you basically are like, you have the image of the person in front of you, and it's like you're passing your hands over the person, and you get this feeling in your hands when you pass over the part of the body that you're visualizing where there's something wrong. And I was like surprisingly accurate at it.
The cat one was craziest to me. That's what I was thinking.
Cat one was insane. So like one of the ones you were supposed to like sit and visualize a pet. And we have a cat, Mingus, and I'm sitting in the room and I feel like this weird pulse in my left eye. And it feels almost like there's this like light that's moved into the room. And in the moment I open my eyes to see if there's anything, and I realize that I'm like sitting in such a way where there's nothing that could have hit my eye from the window. And I'm like, oh, that's really weird. I definitely felt this like very strong feeling in my left eye. And like an hour later, I go out into the living room and the cat's out there and she's got something stuck in her left eye.
Like a little seed pod thing or something.
Yeah.
Man.
And I was like, that's weird.
Yeah.
But I don't know what to do with that, right?
It's like you have this experience.
What do you do with that?
And you walk away and you can rationalize it to yourself all day long. You're like, well, maybe I noticed the cat earlier in the day and I just didn't register it or something, or I don't know. It's just weird because both of us have had paranormal experiences, but it's so easy as scientists for us to just, wash those away and just, there's like, it's so hard to hold on to anything.
It's so hard. And I think that's like what's fascinating too, again, with like what changes people's lives, right? Like an experience like that, but not like for scientists, right? Or like rarely, but it's like, I think that's like kind of interesting though, because I also like, and so I feel like you described what happens, like someone gave you a protocol and you followed the protocol and it maybe worked, right? Or like on some level, it's like an interesting response. And for me, this is like one of my first things to that I didn't share is when I was like diving into the UFO stuff, was like watching these like common documentaries on them. And I found like the documentary on Bob Lazar interesting because I was like, he sounds like me. I trust that he like is interesting. Like he sounds like the things I would want to do in that way. But I was like, there's no there. But what was curious is like the Stephen Greer documentaries. So Stephen Greer is a really divisive figure in this community where he basically like kind of comes off as like a cult leader and kind of like obnoxious or like unaware, but he always like keeps delivering like really interesting things. Like I haven't briefed Bill Clinton on UFOs, but Stephen Greer has, and there's like apparently good like research. Like he shows up in places where like you kind of have to take him seriously on some level because people like keep interacting with him. And so one of his documentaries is like on CE5 contact, which is this thing where it's like if you meditate, you can interact with entities and then like make contact with them, and so you can like invite them to interact with you somewhere. And so it's like, okay, you meditate, like interact with these things, and to me that was like a protocol, right? Again, he like gave people a protocol for them to like do the science themselves, right? And so like kind of with this in mind, I decided like try and do like meditation with the idea of like doing remote viewing. And so I like went to one of these like online sites that there's a tool for this where they have like a picture that's hidden and they give you a number. And so you like meditate on this number again, like that patient's like demographics. And then you have these systems for trying to like get information from that. And it like seems like come really quickly. And so I had a couple experiences where I had like some of those flashes. There was like something I say in my mind that when I looked at the picture, I was like, that just like matches really nicely. Or it's like kind of thing I like wrote down. And it's interesting what to do with that, because then it's like, as a scientist, what I was interested with the UFO stuff was like again like new ideas and technology, right? And so now if I can like remote view and have access to this like crazy new realm of information, like what do you do with that? And I think that's like an interesting thing. And this is actually very much fascinating, I think, with like a lot of secret society stuff is these ideas of like groups of people that do this, that do have like try and remote view or like interact with entities to get like information for technology. But the thing is, again, what you're saying is like, what do you do with this? Well, this information is not necessarily like reliable. Like maybe worked the first time or once, but what about the 10th or 100th or that like and the US government, like you hear these people talking about like describe this thing of like tricksters, right? Like this information is like reliable sometimes, but not all the time. So it's not reliable, right? Or like, how do you like interact well with this? And this opens this whole series of like questions, and it's like an entire like decades of like an infinite amount of like scientific effort to like get there and to like really interrogate that. And so it was like with me was like how I was convinced that like there might be something interesting here to study, but then again, like I can't go to work at my friends instead I like remote viewed a random picture or something, right? That's like it doesn't move us forward, like truly. And this is like the issue with like there's nothing I can like really do with that. And except like what I like want it to be. I think that's kind of the thing is like when people start to have these, this like people tend to expect like you go out and you change your life and you're like win the rand whichever what prize $1 million for proving something like supernatural. And it just seems like that's not actually what happens when people have these experiences. It's like different. So again, like I changed like my own expectation, because now I like had kind of like completed this protocol. I got the result, and it was like the perfect science experiment, right? Someone gave me a list of instructions to follow. I did, and I got a result. It's perfect. But what do you do with that information? And so this is where I think the Christian esoterica comes in, because I think that was throughout this point in my like journey of like interacting with the UFO stuff was in coming across this and being convinced that like there's something interesting to study here, but I'm not quite sure what it is. And there's no real physical evidence or data. So it's hard to get like nuts and bolts information besides just like, well, it can probably move this fast according to a radar operator. It's like a useless bound almost. And so, but there is like these interesting things of like these entities that come and like abduct people. They perform genetic experiments. They share stories. They share visions of apocalyptic Earth. They say the rumors are that like on Roswell there's like human DNA, right, like floating around like the bodies that they recover. There's all this like rumors, and so how does that all fit together? And I found what was interesting to me is when I finally came across like Christian esoterica through like these UFO blogs, was how it was this theory of knowledge that apparently we've like been handed down for maybe thousands of years, if you believe, and that gives us these frameworks by which to understand this information. It's not like we're just like facing this now. It's like there's this entire sect of human history where we pass on these stories about what these things mean, what the like interactions are, and like how this stuff is mediated. Not just like UFOs and alien contact, but much more so the telepathy, the human experience development. Like, what do you choose,
the cosmology,
like, or not?
Yeah,
the
like the cosmology of who we are in the context of all of these other entities and so forth.
Exactly. What's our relationship with them? Right. These kinds of things. So the whole like the water journey, like pull up the wall over my eyes, like I should like interact with everything myself. And then so it's like not necessarily trust and like make my own judgments. And then the UFO journey of that for me was then to be like, we could like miss really important data. And then it kind of like end up here of like there are people who claim to not have been like missing data, they've been like studying it, and that this becomes like these esoteric traditions in some way. And esoteric traditions being like really niche or in depth, like I'm a scientist of like an esoteric pathway of amyloid formation, right? Like a really like niche kind of area, but it's as opposed to like dogmatic or something that's just like a common like church or something. So esoteric Christianity is like a very different kind of Christianity than you get by going to like a dogmatic or a church area. And so those words just describe like a difference in kind of like how these things are presented publicly. But I had come across this link that someone posted in like a UFO subreddit thing to a Gigi Young YouTube channel. This was like fascinating because she had this like series called Esoteric Keys to Disclosure. And it was the entire like all of the like things I've been looking at, like government whistleblowers, UFO abductions, telepathic communication, genetic experiments, human DNA and alien crafts, like what does it all mean? And she kind of laid out this like from this Christian esoteric viewpoint of like her interpretation of this, of had this framework where she placed all of those like nicely with each other. And so it's like
In a sort of
me.
like ancient mythological structure that
That's right.
involves like a whole galactic organization of entities and so forth.
That's right.
Different levels of consciousness and all of
And
this.
it seems like there's also two poles of like the good and the bad and this massive fight that spans all the time.
Before we get to Gigi Young, and I'm actually afraid we need to do an entire podcast about this because this is like too big of a subject for us to roll into this. But
does this wrap back to water?
Yeah, but hold on one second.
Okay.
Definitely.
It reminds me I just wanted to touch on, before we move past the remote viewing experiences that like we've had, is that it kind of reminds me of using ChatGPT. Like it will give you like useful information sometimes, but it might completely lie to you sometimes.
Yeah.
And I almost wonder if there's like a parallel there in what's happening physically with large language modeling, where like you're searching all of this information and you're pulling stuff out of it, but your connections could be wrong, perhaps. Like there might be something there about the searching that's valid, but the output could be deranged and hallucinatory, as maybe in our case as much as not. And so it leads us to doubt the entire experience. But it's like, I don't know, I have this friend, like one of my best friends is always just like, Shiloh, like you've had these experiences. What's your problem, man? Like know it's real. Like what? And I'm just like, it's just the unreliability of it that throws me off. And that's the same thing that I experience when I use ChatGPT too. So I'm like, or Wikipedia is another great example, or something where it's like, this is pretty useful sometimes, but it's also, it's just riddled with mistakes too, and opinions and biases and all this stuff. And so I wonder if there's something about this whole remote viewing experience that is challenged by the cloudiness of accessing information and then articulating it together at the same time.
It's completely. And there's like so much I can respond to about that. Like, people have been doing this for a long time. So kind of like, this is like the CIA ostensibly with their remote viewing program had to like develop an entire series to deal with this, because it's like, when you have a near-death experience, like which saint greets you at the gates, like really depends like on who you are and what you come in with, right? It's like the reflection you bring with you. It seems like people in remote viewing are worried that when they're asking questions, that the person who's doing the interview to get the answers is like projecting their thoughts, and that's what the other person's picking up on. And so you have to be like really careful. So there's this entire like history and methodology of humans trying to figure out the best scientific protocol or methodology to get the best information from this. And this is really like what Gigi Young talks about. Again, like coming back to is that she is a big proponent of this thing, like spiritual science, as she calls it. And a big issue of this is discernment. So how do you tell if ChatGPT is just like telling you something useful or making it up? Well, for me, like I'm asking it questions and I have a PhD in what I'm asking it. Like I like discerning it. But as soon as like I shift that, it's like, I don't know, right? And so there's like, you have to use like this inner discernment no matter what, right? And so Gigi Young talks about this point of like spiritual science. A big part of it is developing discernment, how to tell what truth is to you, right? And so in navigating this, where you are like ostensibly like picking from, and many people do describe like kind of a Freud, like a general unconscious of humanity that you're just like pulling from like randomly, something like that might exist on some level, but also people who seem like really targeted at getting what they want. And an example of targeted at getting what they want is specifically the protocol you're talking about, where it's for healing, right? So are these I don't know about this protocol, but are the visualizations about like a lot of like forming the mind and how you would like highlight different areas of the body as you're like going over it to like get a trigger, right? And so you like build up these things, or these heuristics, or these like things where like purple might be a color that corresponds to healing, or red might be corresponding to something else, right? And so as you get like a flash of a color, you've built your own relationship with yourself then to interpret this data, right? And so again, you're like entering into a world is a totally different like language. And what is odd or like interesting is, again, you're like having a conversation with yourself. You're like telling your own self like what this is.
And the more that you think about it, the more that shifts, right? Because your first association of, let's say, purple healing, red damage, then you start to think about it more and more, and maybe you start to get a different sense of how those things relate. And so over time, and many visitations and many contemplations, all of the sudden, your sense of what red is has shifted, and your sense of what purple
Right.
is has shifted. And so
Yeah.
anytime that you're measuring something internal, you're endlessly remodeling the substrate that you're measuring, because through contemplation, you are fundamentally altering the ground that you are trying to look at.
Yeah. I agree completely. And I think that this is interesting because like I feel like scientists in general, we try and operate from this point of like ground or shared truth of objective reality. And I feel like, maybe an unintended consequence of like the 2022 Nobel Prize or a lot of these things are like uh um uh
The lack
is
of
really
reality.
undermining the lack of reality, right? And I was trying to there's another David had with some common person who uses game theory to kind of like disprove our ability to like handle reality, right? Again, like this is like scientific method is really undermining how much we should trust like the material world, and what it becomes is that we tend to like recreate it as we like are going around, or like when we measure the collapse of like a quantum particle, we're part of the measuring to we're not like remove, we can't seem to create a system where we remove ourselves from, and so in a sense we're always interrogating ourselves, right? Like you are the experimenter looking at that particle. That particle's like part of you looking back at you, right? Like, and so I think that this is like what's cool for the future of science is to really deal with like what that means for our profession, what that means to go into this unknown and try and develop these like ground truths that are useful and reasonable to ourselves or like share across ourselves. And again, this is what like Rudolph Steiner and Gigi Young talk a lot about with like anthroposophy, or these different esoteric Christianity ways of trying to keep this dialogue. And right, this dialogue, it's not only just you that's like interact with you, it's also you interact with human history. So when we have these common concepts of like Jesus or Lucifer or these characters that we hand down, right? It's nice to have a conversation with the past by using these same characters, but also these same characters like change their meaning with time, just as you change your own like relationship to different parts of your life, right? So what does discernment mean in like spiritual science? Like it must also mean that it's like in the right time and place, right? So if you're like to try and communicate with someone in ancient Greece and like talk to them about an iPhone, like do they have any ability to discern the information that you're giving them and turn into something useful for them, or will it just be picked up crazy noisily? Right? What is like the same thing that you have in common with that person? So like you can get into trouble by going out of time and place to interact with this information. And again, like this trickster idea comes through here again, right? Like if you're not interacting with this field in a way that is, sensibly through spiritual science, we've like refined our ability to get useful information. But the whole point is what is so fascinating to me is that there is a whole system of human thought that we've constructed really detailed and over a lot of time that concerns how to extract information in this kind of a way. And now we're confronted by science kind of revealing to us that like maybe there is like this unknown information realm that we do seem to interact with and we get info from it. So like, how do we do that? Like material science is right up against what this esoteric science has been getting at for a while. And so I think that's the power of me like interacting with these stories and taking you trying to like understand like their wisdom and how they're interacting with these, or what are the stories that they worked really hard for this information, right? And so I think that it's worth paying attention to some and trying to see what we like, and also what they're trying to say, not because we do or don't like it, just to understand their entire framework, right? Because it's so alien to how we understand ours, right? And so in this like esoteric framework of particular like Rudolph Steiner, 1910s like Austrian mystic, and he so there's like a short history here of like
all right, so here's what I think we should do. I think we should schedule an entire conversation about Rudolph Steiner and Western esotericism. I've been listening to a lot of these lectures too, these Steiner lectures, and
yeah, we've been listening to them while we cook, and it's pretty interesting stuff because it's ties into the UFO stuff really neatly, and all of the Diana Pasulka stuff and the like ancient philosophies.
And there's also this real sense that we've had, like the further we've been into science, especially just talking to all these different scientists on the podcast, the more interesting it becomes to me that not having any kind of spiritual grounding in your inquiry is impossible, right? Like whether you're going to bring a metaphysical stance to your investigation, whether you realize it or not. And it seems to me that the assertion, at least from Gigi's lectures that I've listened to, is like, that we should be aware of that. So the idea of like a spiritual science it sounds kind of woo, but like it's more just like taking control over that, taking responsibility for that metaphysical bent that you bring to an investigation in the first place. Really being aware and at least incorporating that in, because I feel like scientists today, it's really obvious to me in cosmology and astrophysics, but like your belief structures inform your theory constructions. And if you pretend like you're objective and you're clean of that being possible, like you're lying to yourself. And so it would be probably better if we were just outright and open about our metaphysical bents. And maybe we should cultivate them too for the best possible outcome as well, if we're going to acknowledge them. So I just think I would love to just sit down again and actually unpack some of these mythologies with you and how they relate to the modern day, because that sounds like a really fun conversation for me.
Sounds great.
Can we wrap this back to water? Does this wrap back to water? Because that's where we started, and I love the idea of like a neat package that has a bow on it.
Yeah. For me, I like a lot. The best way I can have is that I think that water is like in these traditions is like medium, and again this medium where like things come through, right? Or like it is kind of using rituals in a way, or and I think that for me, I kind of fascinated by this idea that this interaction between light, water, and your like body, or the proteins, the molecules of your body. And so like the esoteric thing, or like people in these like woo new age things will say that we're like beings of light, right? And at the heart of that OR thing is how these microtubules are like nicely poised to be this like transform of light information into like physical motion information, right? And so for me, I think that, and what's like really useful about water as being this like transfer of it is that it's so sensitive to so many electronic moments and magnetic moments that's going on. It's so exquisitely sensitive to its like really local environment, but also to grand like changes, right? And so there's this effect called the Scholl effect in biochemistry, where this very, very careful Swiss chemist thought that like this chemical reaction, these peaks were changing their height all the time. Like, and you notice there's like different times of year. And so we try and like very carefully measure when his like peaks are changing, and then he's like comparing this like motions of Jupiter, right? And so again, like how this like extrapolates out is that now if you're exquisitely like electrically and magnetically sensitive with your water, that you can be picking up on all these kinds of, again, this basically ties in directly to like astrology, like giving this a physical mechanism by which we like connect. So the black hole at the center of our galaxy has got like a magnetic moment, and that flips occasionally potentially. And then our sun has a magnetic moment that flips occasionally. And then our Earth has a magnetic moment that flips occasionally. And all the atoms in your body, or not all of them, but like many of them do, right? You're filled with these, right? And so there's some hierarchy of like communicating up and down through these like master things. Again, Michael Levin's talking about this, like electricity is this master regulator. And so how this like ties in with light, and it all comes together, like water is that like interface between these two, right? Yeah.
And it's such an interesting example of something once considered to be passive containing a great deal of active potential. Like it's like, it's almost like I look at the search for the ether back at the turn of the 20th century, and it's like they're treating the ether as this passive like backdrop, right? And it's like so many of these experiments start to make sense once you consider them really, the ether not really as distinct from the atoms, but just as like extensions of the atoms themselves. And you actually have an active structure now, because the ether, whatever it is, the medium is just as active in doing all of the actions that we traditionally ascribe to the atom. And so just this idea that these things that we assume as passive backdrops could be huge, if not the central players in the stories, I think is really how this all wraps around back to the beginning.
Yeah. And I mean, I think that that's the arc of how science has developed anyways. If you look at the way that they talked about DNA 30 years ago, they're like, man, there's a lot of junk DNA that's not doing anything. Mm-hmm. And as time has gone on, there's gradually been this realization of like, well, actually it does seem to be doing a lot, but we have no idea. And so it's the same thing with water. It's the same thing with the extension of atoms.
Microbiome.
It's just we have no way of dealing with the void. Like we see the void and we're like, it must be black. There must be nothing in there.
Right.
And then we start to put up lights and we're like, oh shit, it's not actually a void. There's like a bunch of stuff in there that we just hadn't seen before.
Yeah, I always come back to the fractal coastline problem of like, what's the coastline of Britain? It's your ruler. If it's a foot ruler, it's measurable. But as soon as you make your ruler smaller and smaller, you have to go around like every pebble on a beach on Britain, it becomes infinitely vast. And yeah, the curse of our amazing attosecond lasers is that we can measure a lot more of this like surface area of the void. And yeah, it's so much information. It's a struggle to know what to do with it. And I think that's like what's in front of us. Like, how do we narrow our search space to get what's useful to us? I think that's like a really important
I love
thing
that.
to keep in mind.
The
Yeah,
frame is the story. How is it
right,
useful? What are the things that we are going to discover, and what is the frame on which they fit that is not yet the dominant frame? Because we
right,
know that
right.
there must be a new one, because that's what happens to science. It
Yeah,
moves to a place where we're like, oh shit, all this stuff that we didn't see before is actually there.
right.
And the only way that we can see it is by following the path of somebody like Levin, who's like, okay, well, what if the philosophy that underpins this is completely different from what we thought?
Bingo. And we can choose any framework because we're going to invent it. And so it might as well be spiritually informed if we want, or like it could be, right? Like there's all this like room for
Yeah.
we invent
Or
the framework. And so what do we want this framework to address? And
Mm.
its utility, what's useful to us, right? Like
that's like the
uh,
conscious possession of the spirituality. It's like it's gonna be there whether you recognize it or not, I guess is
right.
kind of the way I
So
see
grab
it.
hold of that power, right? It's yours whether you like it. If you don't wield it, it's being wielded for you, right?
Right.
And so as
Right.
scientists to consciously take hold of this, like now that we're aware, the future is bright and bold if we choose it to be so, right?
Hell yeah.
I love that.
All right, man. Well, if you're down, let's do this again soon, as soon as you have
Yeah,
time or whenever,
absolutely.
and as soon
Yeah,
as our schedules align. Because I want to. There's so much. I really want to unpack this stuff because I don't have
yeah.
anybody else to talk about it with, and we've been
I
chasing down this trail, and it ties into so much of the mythology that we're interested in, and the way that humans have understood their place in the cosmos seems, in some sense, better informed in the past than in the present, right? This
yes,
Big
yeah.
Bang cosmology just seems like completely vapid compared to these stories that really give meaning to people's lives in the past. And so maybe we can just start there next time.
Let's start there next time. That sounds great because again, that's what's cool to me too, is like the meaning of those stories transported to like modern day from what I've lost with our modern cosmology is like perfect stories. I would love to pick this up, yeah, whenever we find time. Yeah.
Amazing.
All right, cool, Michael. It's always fun to hang out with you, man.
Yeah,
So
thank you
it's
so
been cool.
much.
Thank you so much.
Yeah.
Take care, guys.
We'll talk soon.
Sure do.
All right. Bye, everybody.
Bye.